आइडिया अच्छा है बहुत
पता नहीं ये पता है उनको मिला नहीं उनको बहुत डाटा नहीं मिला सॉरी मैं तो लंबा मेरे डेटा एक कंटेन है
एक ऑलरेडी है तो
Anyone joining on YouTube? Meta?
Extra बहुत
सर
अभी इस टेबल के आसपास नहीं आना भी कोई क्योंकि ये कॉर्नर नहीं है तो
वहाँ से जाएंगे डायरेक्ट
अभी जी
सो नीचे दबाएंगे तो अपनी तरफ आएगा तो वो बन जाएगा
1 2 3 4 का डायरेक्शन
करोगे ना तो नीचे आएगा और ये राइट लेफ्ट ज़्यादा ऊपर नहीं करना वो वापस ऊपर का मत करो
am I audible? Am I audible to you? Can you switch back in? Okay.
ये पहला मैं ऑन देता हूँ थोड़ा तो हाँ सॉरी
Now window.
लाइव प्रॉपर
Audio video.
हां
1 to 9 हो गया अब हम लोग भाई देख इसका है ना इसका साइड वाला तो बताना नीचे ऊपर वाला ज़्यादा है उंगली मत करना
नहीं मुझे वो काम है
ये sensitive. ये नहीं
तेरे को तो प्रेजेंट करना है
अच्छा इस पे डिपेंड अच्छा स्पीकर पे
बाद में यूट्यूब पे आ रहा है एक बार
हॉस्पिटल को
आज आ रही है
Hello everyone. Hello everyone. Is from group around so that we can start.
2 online I think
Can
2
you
online
call your collaborators so that we can start the
chatShaala का
event?
लिंक चाहिए so chatShaala का लिंक ये दो कोलैबोरेटर्स ऑनलाइन ऑफलाइन हम 2 ही लोग हैं और ये तीसरा लिंक शेयर
अच्छा सेम लिंक पर था ना
Join करेंगे.
पानी पी लीजिए पानी का अरे हाँ यहाँ पे पीछे क्योंकि पानी बहुत बंद हो जाता है, सबको टेंशन आ रहा है तो पानी लगेगा पीछे। नहीं तो आपका नंबर जोड़ लेता हूँ, आपका तेरा नहीं आया तो आपका है, एक मिनट। तुरंत कितना फॉरवर्ड कर दीजिए। हाँ
साक्षी फोटोज पाठव. सगळेच पाठव. जेवढं आहे ना
साइड वाले साइड वाला है नीचे वाला की ऊपर बट ऐसा लगेगा येलो वाला
Hi Vivas, welcome to the second day of Youth National Meet Vivas. You can also switch on your web camera. We will be able to see you on the screen. Hello Lahari. Lahari, are you around? Can you switch on your web camera?
रेड भी नहीं किया कभी दीवार से करता जैसे रेड भी नहीं किया कमरे को कुछ हेल्प करता नंबर से नहीं हो रहा
Yes, Lari, we can see you. Good to see you around. Ashrita, can you also take the mic? You are on listen mode. Is there anyone going to join online from Atman group?
देख रहे हैं भेज रहे हैं
Sahin.
So hello all, good morning and welcome to the day 2 of Cube National Meet 2023. As the Somaya ritual goes, we will start the day with the Somaya campus prayer.
पूर्णमिदं पूर्णात् पूर्णमुदच्यते पूर्णस्य पूर्णमादाय पूर्णमेवावशिष्यते ॐ ईशावास्यमिदं सर्वम् यत्किञ्चत्यां जगत् तेन त्यक्तेन भुञ्जीता मा पृथक्तस्य पृच्छनम् मूकं करोति वाचालम् पङ्गुं लङ्घयते गिरिम् यत्कृपातमहं वन्दे परमानन्दमाधवम् ॐ प्रणो देवी सरस्वती वाजेभिर्वाजिनीवती ीनामविद्यवतु ॐ चोदहि सुमतीनां यं दधे सरस्वती ॐ असतो मा तत्समय तमसो मा ज्योतिर्गमय मृत्योर्मा अमृतं गमय ॐ सहना भवतु सहनौ भुनक्तु
Thank you all. Now I will just give you a list about what all things we discussed and what all presentations we had yesterday, and then we'll go to follow with today's program. Yesterday we started with food fly group with amazing discussions on topics like diabetes, which how students through this are developing just by discussing in chatShaala, just through collaboration. Other systems like activity pattern, which are to be discussed in a sense where students themselves develop a habit of creating such awareness among people that even if they go to their kitchen, they will see fruit flies, they can take photos, and they can be part of the activity pattern group. We discussed about the other possibilities of research topics in Drosophila, that is Alzheimer's and uh, transgenic species, and how Benzer's contribution is there in Drosophila model system. Then we went ahead with our second presentation, that was microbes. In our 11th standard student has worked enormously well in this field, and the contribution with the chai wala microbe is to be acknowledged, that people from Jammu and Kashmir, the normal people who their daily life depends upon all these things, they want to collaborate with INAS and do things. Then we went ahead with our nematodes presentation. We discussed about the search of native Indian soil of C. elegans, and how we are going to develop it as a model that not just C. elegans but also let's track whatever we are getting, and let's try to develop it as a model system, and let not just wait for the gold till we have silver in our hand. We went ahead with cardamine presentation. They discussed about the very simple method of genetic engineering, how a floral dip method developed by some people at Max Planck Institute is helping cubists to understand their curriculum better. Curriculum in a way where they are attached to the topic, the syllabus is uncovered and not covered. So next we went ahead with butterflies, a beautiful model system. This plus Sara's contribution from Thiruvananthapuram and Tirtha's contribution and Tirtha's energy in butterfly model system that she expressed yesterday. I think the best part of yesterday's was the dog model system. Everyone's favorite. And we discussed about how we can this very simple organism which we see every day around us, how its daily activity pattern can be tracked. Is it irregular? How it changes with age? How it changes with the condition in which the animal is placed? Either it is a domesticated one or a stray one. What kind of behavioral changes we can see in dogs? And we found out a possibility of connecting it with humans. Thus, sleep and this pattern changes in humans as well. When we grow old, when we are young, and when we are doing anything for that matter. So today, we are going to go forward with our today's flow. But before that, I would like to introduce you to our today's chairperson for the first, Sushanta sir from Q Assam. So Sushanta sir has given enormous contribution from Q Assam. Like he has inspired many students from there to join even the one interesting story which Aruna sir always refers to is last time when sir came to Mumbai, and when they were going back, they captured the photographs of mango trees all along the way, and they created a map of their paths, which we still today have with us. And I think the Seasonics group is going to show that contribution by Sudhanta sir. This time as well, he brought to us a gift of philanthus plants from Assam. So how this culture of playing science every day and everywhere that we are going is inculcated by people like Sushanta sir. Sir, please welcome. I welcome you to the stage, and he will be chairing the session for us today. And let's go ahead with our presentations.
Thanks everyone. And Stella. Good morning, all, and by heartily welcome to again to the second session of these presentations. Yesterday we have amazing presentations over here, and now our organizers give us handed over the schedule. This very they have given a hard and fast rule for today, and so we have lots of presentations to be conducted here, and I think we got 9 presentations before lunch. So we have to keep in mind that we have not so much time like yesterday's sessions where our discussions were going to larger extent, taking much time. So we try to finish every session within 20 minutes. So thanks everybody for coming here, and I'm instructed to carry out the sessions. Now I'd like to request the alpha group to the dais for the presentation.
Okay, so good. Yes, yes. Hello. Can you hear me? Loud and clear? So, good morning to everybody. I am Vatsal Savla from KJ Somaya Cube Lab. This with us is Abhijit, as everybody knows. This guy is Mohammad Awais from Ratnam College, Bhandup, and he's with us in the cube. And
I'm Bhupati Sahu from Chaitanya College, Palmer, Chhattisgarh.
so, as you can see, our model for the cube meet is earthworm. So, there must be a question like we see earthworms every day. So, why earthworms? So, the reason for that was that first of all, they are very easy to find, especially in monsoons. They surface up because earthworms, though they need moisture to breathe, but they don't like too much of water around them. So, when the water seeps into the ground during monsoons, they surface up so as to not suffocate. Secondly, they are very easy to culture and maintain compared to other model systems that could be in the Annelida family. Third, earthworms have a cerebral ganglion, which is very similar to a human brain but very primitive, so it's easier to study it. Fourth, they have a PNS and CNS, like a peripheral nervous system and central nervous system, which is having some similarity to humans, of course. And fifth is both earthworms and humans have a closed circulatory system. Means it's not open, it's closed inside the body, and we will move on ahead.
actually this laptop is lagging. So, are all your collaborators around? Is there anyone also going to join on Big Blue button? No. Give me seconds. You can talk something about that one. It is actually low.
The problem is that computer is lagging.
Okay, so I think we are facing some technical issues. So while it loads, I'll move on with the presentation since we are running low on time. So while studying earthworms, we came up with a few questions. All our collaborators came up with a few questions and clubbed them together. First one was, how do different culture methods affect earthworms? Because naturally, if we found them in soil or in rocks where there is access to soil or moist areas, but as we have observed, we can culture them in different media. Let's say newspaper, let's say tissue paper, let's say fruit peels. So how do these different media or these substances affect earthworm culture? Second was, what are the factors responsible for ventral cord regeneration? So as we know, earthworms have a ventral cord in them, and yes, so it's a no. So earthworms have the ability to regenerate, as we know, but how does it affect the ventral cord, or the ventral cord affects regeneration? Third one was, what are the factors responsible for the ventral cord regeneration? Just as we discussed right now. Wait, this is COVID. Okay, anyways. And the third one is, what is the role of clitellum in regeneration? So first one was ventral cord, now it's clitellum. So as I just discussed right now, types of earthworm culture. So I'll give Mohammad Owais to continue.
Good morning, everyone. There are three types. Myself, Mohammad Owais. I'm from Randham College. So I'll be speaking about the cultures of earthworm. So basically, there are three types of cultures in earthworm, which I have carried out in my setup. One was soil, tissue culture, and the fruit pea. So in my setup, I've taken the first of all, I've taken a beaker in which I have put some soil, which I've got around my garden area. In the second culture, I've kept some tissue paper. I've placed some tissue paper pieces, and I've again placed the earthworms in it. And the third was the fruit pea. So first of all, my aim, my objective, or the aim which I can say, was to observe that whether it can survive in the different conditions, and what more it can do in it, like whether it dies or regeneration happens or reproduction happens. So these 2 to 3 basic things was my aim. So the first was my tissue paper culture. In tissue paper culture, first I talk about the pop-ups which I've made. After observation, first thing where Arunan sir corrected me, I was the first time when I tried out in tissue paper. I knew that the paper absorbs the water, so I did not put the excess amount of water, but I did not put enough water. I just sprinkled the water upon it, so that I knew that the water taps inside it. So I thought placing the earthworm would be fine. But it eventually got evaporated after a few hours, and I kept watering it regularly, but not that much how much it needed. So within a week, I found out that my earthworm died. So the reason my earthworm died, because the water is evaporated in my tissue paper culture, and the moisture which earthworm has on its body has also been evaporated by the tissue paper. So I kept this problem in front of Arunan sir. He corrected me. He pointed out my mistake, that water has to be the water has to be put in, not in excess but not too low. The water should not be sprinkled on it. The water has to be put that much that the earthworm gets its enough moisture, and it does not die. So one mover and one means how to do it. So I just thought that putting a peel is enough for it, because I've chosen, as you can see, papaya peel I've chosen. I thought it has stickiness on it, so moisture would be there. But it was my mistake. I accept that. I did not put water, neither the soil, because I did not knew. I was the first time. It was the first time when I tried it. So after few days, as you can see, my earthworms has been dead, found dead, and it has started to rot. So after I again asked sir that what was this issue, then he again pointed me out, and some of my friends like Vatsal, he also helped me that he pointed out my mistake that the soil, which is a substrate in its food, has to be put in it. Just the fruit peel was not enough because they survive on the dead and decayed matter. So they do not eat it. They suck their through their organ pharynx. They suck the nutrients and the portion which it needs. So my peel was fresh, it did not start to decay. So it will take time. So until that time, it will go. So this was this was one of the second mistake which I found.
So as you guys can see, this is our earthworm culture of Somaya lab. So we set up this culture on 1st of July of 2023, and we used a big tray, filled it with a few layers of newspaper, added water to it, and then we placed some plastic containers that have very fine holes from bottom and fine holes on the lid. Right now we don't have the photos of the lid, but I will. So what we did was we placed cutouts of newspaper into the cup and we added water just to keep them enough moist. So that if we added more water, the water would come out. It would not be absorbed. So the maximum capacity of the absorbance water was put in, and we placed about 59 earthworms in all of these 5 cups. So the corner cups, as you can see, had 8 earthworms each, and the central cup had 27 earthworms. At the time of collection, we had some other plans for the excess ones, but we are still working on it. And after a few days, like on say 4th or 5th of July, we observed that these earthworms were there. Labeling of the cups was done on the lid. So when it was collected, how much worms were collected, and from which location. So even if we do mislabel, like if let's say we have the positions recorded, but if somebody let's say places this cup to a year, still the number of the earthworms would be same, but just the different earthworms. So the number would not be changed. Labeling is to that we don't misplace them. Yes. So I think we will. No, it is very important. Yes, the main I think one of the reasons was lids are easily visible from the top. Like when we enter the lab and when. Okay. No, we are not sure. Yeah, but
That's a very good question from Arun sir. But as we have shortage of time, I'd like to.
I'll answer your question later, and we will work upon the labeling system as well. So thank you, Arun sir. So after a few days. So after a few days, we just for answer like we had a question that whether the earthworms are of different species or whether we are just juvenile and adult earthworms. So we took out 2 random earthworms from cup number 2 and we placed them in the petri dish. So right now, as you can see, we observed 1 adult earthworm because the clitellum was very visible, and we had a juvenile earthworm. But still, we are struggling to find the exact species of the earthworm so as to we can create more better results or data. So as you can see in the newspaper, they were thriving very well in our culture. So the species or the castings were observed within a few days, like day number 2 and 3. And after 14 days, we have observed cocoons as well. This is the same cup that I have shared in the context to curriculum as well. Now I will pass on to Bhagwati ma'am.
Good morning, everyone. मेरा 2 एक्सपेरिमेंट था। मैंने अर्थवॉर्म टिश्यू कल्चर किया एंड अर्थवॉर्म न्यूज़पेपर कल्चर। मेरा ये एक्सपेक्टेशन था कि अर्थवॉर्म टिश्यू पेपर एंड न्यूज़पेपर में सर्वाइव कर पाते हैं कि नहीं कर पाते। ये एक्सपेरिमेंट मैंने स्टार्ट किया था 15 जुलाई को इन माय कॉलेज एंड मेरे साथ मेरे स्टूडेंट भी थे जिन्होंने सेटअप में हेल्प की थी। तो हमने स्टार्ट किया था। हमने टिश्यू पेपर कल्चर फॉलोइंग दिस पिक्चर इस टाइप से किया था हमने। सेकेंड डे हमने पाया कि जो टिश्यू पेपर में हमने अर्थवॉर्म कल्चर किया था वो सेकेंड डे सरवाइव किया था। नेक्स्ट स्लाइड। दैट आई फाउंड हमने सेम डेट पे अर्थवॉर्म न्यूज़पेपर कल्चर भी किया था, लेकिन हमने रिजल्ट ये पाया कि जो हमने न्यूज़पेपर में अर्थवॉर्म कल्चर किया था वो डेड हो गया। हमने अर्थवॉर्म टिश्यू पेपर कल्चर में 1 अर्थवॉर्म लिया था, लेकिन न्यूज़पेपर कल्चर में हमने 2 अर्थवॉर्म लिए थे। लेकिन हमने पाया कि जो हमने अर्थवॉर्म कल्चर कर रहे थे वो उसका डेथ हो गया था। तो हमने थोड़ा सा सोचा कि इसके डेथ होने का क्या रीजन है। तो हमने हमने एक कंक्लूजन निकाला कि न्यूज़पेपर बनाने के लिए बहुत सारे केमिकल्स का यूज किया जाता है। बहुत सारे इंक का यूज किया जाता है इसमें, जिसमें सबसे हार्मफुल जो रहता है वो थैलेट्स होते हैं, जिसके कारण अर्थवॉर्म सरवाइव नहीं कर पाया होगा और उसकी डेथ हो गई होगी। लेकिन बहुत सारे कोलैबोरेटर्स से बात करने पर यह पता चला कि वो जो न्यूज़पेपर में कल्चर कर रहे हैं वो अर्थवॉर्म सरवाइव कर रहा है। तो दैट हमने ये सोचा कि हमसे कुछ मिस्टेक हुआ है जिसके कारण अर्थवॉर्म सरवाइव नहीं कर पाया। दैट फ्यू डेज हमने स्टार्ट किया था 15 जुलाई को एंड 18 जुलाई टिश्यू पेपर अर्थवॉर्म कप कल्चर। तो अर्थवॉर्म अभी भी सरवाइव कर रहा है और अभी तो हम लेकर नहीं आए हैं, लेकिन टिश्यू पेपर में जो हम अर्थवॉर्म कल्चर कर रहे हैं उसको मेंटेन एक स्टूडेंट कर रहा है और वो अभी भी अलाइव है। This is picture is 19 July. Earthworm is alive and survive.
As we had mentioned in our research questions slide that there are few questions that we wanted to address, and one of them was how regeneration takes place in earthworms. So, for example, when humans are getting injured in their spinal cord, let's say for our understanding that we are not able to move our body below that part, or our body gets paralyzed. Earthworm, in the case, a very smart organism, is able to get that part back. It's able to regenerate that. So what are the factors which are responsible for this regeneration and how we can study it? So cubists developed some ways through which we can study it very clearly. So we can go ahead with the slide and we will be able to see that cubists, when we were discussing in chatShaala, not only chatShaala but before in the offline causeries when the cube used to happen in Homeava Center for Science Education, the discussions जो वहाँ पे होते थे, so वहाँ पे cubists ने एक method develop किया to study the ventral nerve cord regeneration of earthworms. So ventral nerve cord जो है, it is same like the spinal cord of humans. Just in the earthworm it is present on the ventral side of the body, इसलिए it is called as ventral nerve cord. वो starting segment से लेकर it goes till the end throughout the body of the earthworm. So you can see here, if you take this earthworm, this blue line you will see is the ventral nerve. So that you just had a question in our mind that if the ventral nerve cord is cut, then what will happen? And how do we test that the ventral nerve cord is cut and the earthworm has lost its coordination? And how do we again test when it gets its coordination back by regenerating the nerve form. So then the cubists came up with an experiment which they call it as rapid escape response test. As this word suggests, rapid escape response. So there are three terms which I have to talk to you about. So what they did is, when we take an earthworm, or let's say we are putting the earthworm in a salt solution, we expect that the earthworm will run away from the salt solution because it breathes from its skin, and when the salt come in contact with their skin, the cells start to deteriorate and the earthworm will die. So earthworm has to very quickly show that कि उसको सट से बाहर आना है. The earthworm has to show very good coordination that it has to come out of the salt solution. So for that they designed this experiment. So what we did is, we took test tubes. In one tube we will take normal water or distilled water as per the availability. We will put the earthworms inside the tube and we will see for how much time the earthworm stays there and how much time it will come up out of the water. And the other part, we will take salt water of desired concentration and as per the available things in our home lab. So we will make a salt water solution, and if we put the earthworm in that solution, then how much time the earthworm will come out. Doing some actions but not that quickly coming out. But when we, on the other hand, we see the video of NaCl, then we will see how the earthworm is escaping through the walls of the cup. And this cup wall is very slippery. So the earthworm has to use all its energy to come out of the salt solution so that it can survive. Now let's say the same particular earthworm, we have given a lesion on the ventral side, which we think goes deep to the ventral nerve cord and not the cutting the earthworm in two parts. Just the ventral nerve cord is cut. Now what we expect, now the coordination in the part where the lesion is given, the cut is given to the ventral nerve cord, is lost. So the front part and the back part has no communication with each other. So when we repeat this experiment in distilled water and in NaCl, then we will expect that distilled water again will have no issue or like that. But when in NaCl, the earthworm won't be able to come out because the coordination is lost. So it will not be able to climb the walls like it is doing now. And when we go on testing the earthworm for consecutively alternate days, like not just harming the earthworm every day doing this, because the salt is very heavy for the earthworm. So as we consecutively proceed with our experiments of rapid escape response, we will see that when the earthworm will start to gain its coordination and again climb back the walls of the cup. So this is the way cubists have designed to test how regeneration takes place in how. So this experiment helps us and tell us how much days it will take for an earthworm to regenerate, when coordination is lost, when coordination is gained back. And this has to be done in multiple steps so that we have a good amount of data, good range that in between this period to this period the coordination is back when the one nerve cord is regenerated, and then we will study it further. We can go ahead and next slide. Now this part, Misbah, our collaborator from Elphinstone College, was also not able to join with us. So this work also she has done in home lab, which I am to present it for her on behalf of her. So here we are talking about the body regeneration of earthworm. So let's say हमने earthworm को दो part में cut कर दिया. So how much time it takes for the earthworm to regain the lost body parts, which are cut? So the head part will not have will have the clitellum. She wanted to test that what is the role of clitellum in the regeneration and like that. So this part is with clitellum and this part is without clitellum. So when we go ahead, we will see that when she cultured this earthworm further in the same tissue paper culture, this is the earthworm one head piece. So that's how we trace it. Earthworm one tail piece, the E1 PP. That's how it goes. This is the earthworm one, so she did it with 3 earthworms of head piece and 3 earthworms for tail piece. So here in earthworm one, these two, as you can see, this is the day one, sorry, earthworm 1, 2, and 3. So you can see how the earthworm here is there, but now it is ready to grow its tail. And here this is the tail piece. So we can see how the tail piece has started to form a little. I hope you are able to see this with small, but here it is prominently visible that it has started to gain the lost aspect. So now it's very difficult for this earthworm particularly because it has lost its head, where the mouth is there to eat. So if it is not able to eat, then how the earthworm is able to regenerate this part back? Because it takes a lot of energy. First of all, the first thing is when the earthworm is cut, there is a wound. So it has to heal that. Comes to immunology, or the earthworm is facing the bacterial and other problems when the earthworm is cut. And the second part is how the earthworm regenerates when it is not able to eat. This earthworm is able to eat because its mouth is there, but the tail piece is not having a mouth. So let's see what happened further. So this is day 0, like that she has done this. So we can see here. So this is the part where it has started to grow its tail. This is day 16 and this is day 22. So we can see how the segments are growing back. This is the head piece of earthworm 3. This is the tail piece of earthworm 3. Unfortunately, the earthworm 1 and 2, they died within the next 3 days. That's what she found in her culture. The exact reason we are still trying to find out, that she was not able to exactly get why the earthworm died when it had its head intact, when it was able to eat. In the tail piece, you can see it is day 0, day 8, it has started to form a structure and then segments are coming. We can go ahead, please.
Abhishek, please hurry up. It's shortage of time.
So this is the question that she was trying to address, and she was referencing all the articles and all that. What is the role of collateral in regeneration and how it affects? She was going through some articles which we have put here. The amputated segments have an intact collateral, they're able to heal the wounds. Because so one reference was in the collateral helps in regeneration. So that she wanted to found. We go in. This is still
So while running the test in our Somaya Cube lab, we had a few questions regarding the gustatory response of the earthworms. So if in very layman terms, I'll explain to you what is gustatory response. So as humans, we have taste and we have the memory associated to it. So let's say I am eating a bitter gourd, it's very bitter, so I don't like it. And if there is I would not choose to eat it. Same happens with earthworms. They find food via olfactory sensors or cells responsible for olfactory. So they can also test that which food is suitable and not. So for our experiment, we use some salt solution and some tap water. So the question comes, why do we need to study gustatory response in earthworms? First of all, as I explained earlier, they have a ganglion similar to us. So their response to taste could help us understand how humans work, but on a higher level. So how are we checking the response? So we first took a text experiment. So in test experiment, we laid out a base. After that, 2 strips of tap water strips, like parchment paper soaked in tap water, and we let the earthworm run in between, so they could pick the side they want to go to. So once the earthworm totally goes to the paper, we concluded that either it's choosing the right or the left. Now for the test experiment, what we did was we took the same base again, and now we took, sorry, sir, we currently don't have the exact video. We have the video, but it is not very good. we are not able to upload it as well. So, yeah. But we'll be sharing it in the WhatsApp group. So coming ahead, during the text experiment, we had now one strip of parchment paper soaked in tap water and other in salt solution. So what we found was that out of the 5 earthworms, 1 adult and 4 juvenile earthworms conducted on 14th of July 23, 2 earthworms went to the distilled water parchment paper, 2 earthworms slightly went to the salt solution, and 1 earthworm just kept wriggling around in between the space that we have provided. So what we concluded was that the 2 earthworms that had their senses intact did not choose to go to the salt solution as it was irritating them, and for the other 2 earthworms that slightly went to the salt solution might have found that the solution was isotonic to their skin, so the salt solution did not harm them, but otherwise they were also flinching in the video. So what do we learn from the observations? That there is some factor that is helping the earthworms to sense the taste of the food nearby, so they can make a quick response whether to eat it or whether to avoid it. Next slide, please. So during our cultures, we made some goof ups. As Mohammed said that he made some goof ups during his first run of set culture. So lack of regular maintenance cultures like hydration issues and the soil, this not providing enough substrate or decaying matter for the earthworms to eat. And second one, in our culture on 8th of July, there was a slight mishap that one of the cups had a loose lid. Now we don't know the exact reason whether we left it or someone tampered with it, but the earthworm escaped. 4 of the earthworms escaped. We regained 3 of them, but one was found dead in the Moina tank culture. So that was a very bad day for us. Moving ahead. So does anybody have any questions?
Thank you. I think your presentation is over. Indeed, it was a very good presentation to the first session. Thank you, Vatsal, Mohammed Awais, Abhijit, and Bhagwati. And we'll have only 3 questions now, as we're running short of time. The first question is from Vivas. It's online, and other 2 will be offline. So Vivas is asking, how is Vatsal's hypothesis is setting up?
Okay, so we have prepared the containers, and after the meet, probably we will be setting up the control for first the controls, and then we are also arranging the processed food for the earthworms. So the set culture will be provided before 27th because we have exams. So.
Okay, and next question is, there is a discussion over here that regarding the cultural newspaper.
Yes.
The question is very important for us also. Was there any effect of ink from newspaper? This is for Bhagwati.
हमने जो न्यूज़पेपर में अर्थवॉर्म कल्चर किया था, वो हमने पाया था कि अर्थवॉर्म थर्डे में वो डेड हो गया था। तो हमें लगा कि न्यूज़पेपर बनाने न्यूज़पेपर में जो इंक यूज किए जाते हैं वो अर्थवॉर्म के सर्वाइवलशिप के लिए हार्मफुल होगा, और सो दैट सबसे हार्मफुल उसमें जो केमिकल्स यूज होता है वो थेरेट्स है, जो कि अर्थवॉर्म के सर्वाइवलशिप के लिए बहुत ही हार्मफुल है।
But you have said that other persons will the same setup.
Yeah. बट हमने कोलैबोरेटर से बात किया तो उन्होंने कहा कि हम जो न्यूज़पेपर में अर्थवॉर्म का कल्चर कर रहे हैं वो अलाइव है, तो हमने सोचा कि हमने हमारे मेंटेनेंस में कहीं ना कुछ गड़बड़ी है जिसके कारण अर्थवॉर्म वहाँ पे सरवाइव नहीं कर पाया। एज दिस ये हो गया होगा कि जो हमने न्यूज़पेपर लिया था उसमें जो वाटर का जो अमाउंट है आई थिंक ज्यादा हो गया था जिसके कारण अर्थवॉर्म सरवाइव नहीं कर पाया।
So any more questions from audience? Please. किसी को पूछना है?
My question is for Abhijit, sorry.
what was the concentration of that initial solution, or you made it roughly?
I don't want.
Very good question for QS.
So actually, when the rapid escape response test was done in the lab, we need to find out at what concentration we need to do the experiment and what earthworm can tolerate. So my senior Gaurangi, she was not able to come today. Their group did an experiment where they tried to find out at what concentration the earthworm is able to tolerate. So they were taking it in terms of 100 millimolar, 200 millimolar, 300 millimolar, millimolar. So they found out that 200 millimolar is the concentration which is good to test the rapid escape response test. But when we talk this about in home lab, how do we make the millimolar solution? So then Iram, she decided that why not let me take 1 liter ka bottle and I will add 1 spoon salt. If the earthworm is giving me the response, that is enough for me. So what is the objective of the test is to see if earthworm is able to escape or not escape. So if in 1 spoon in my home lab it is able to give me response, then why should I shouldn't go ahead with it.
So you don't have weighted that salt. Just give me a spoon.
Yeah, 1 spoon in home lab.
Okay. Just thank you, Abhijit. Any more question? Last question. Somebody want to ask any question?
You cut that earthworm in two parts. The clitellar region is responsible for regeneration. Have you concluded that?
So
If we make three parts, what will happen?
yeah, so that was an interesting question. So what happened was Misbah at that time really took replicates of her setup. So she had 3 earthworms only, and out of which 2 died. So we got exactly so the 2 earthworms died, which were of the headpiece which contained the clitellum. So she couldn't when after 3 days the earthworm died. So she was not able to continue, and only 1 earthworm left, which on the I think the 15th day it left the cup. Like it escaped through the cup. So that is the goof of that the cup was not tied properly with the cloth or something like that. So the earthworm escaped. So not having replicates is one of the goof ups, and not the cup that we have is not. The other part of the question that if we cut in 3 parts, what will happen? That she was in her future plans. She told that I will just first find out that what will happen if I do it in 2 parts. Yeah, so if we cut in 3 parts, then she was again expecting that when her question was that she want to test whether clitellum is responsible or not. So she was expecting that the clitellum wala part, the head part with the clitellum, it will regenerate. Middle part and the tail part might not be able to regenerate as in comparison to the head part which contained clitellum. That was her expectation, but she didn't follow it up later on for some reason. So I think now it is opportunity for us to continue on that.
Thank you, Abhijit. And these are very good questions. And we can have a hypothesis that if we cut an earthworm into 3 pieces, the middle one have cut the both sides. So they might need more energy to regenerate that part. So we can continue on that also. And now, again, thanks you all. And we'll go for the next session. So it was a very fantastic presentations over here with a very small creatures like an art form that have a ganglion like us, and the simple model system will lead to very difficult questions. Answer very difficult questions on neuroscience also. Thank you all. And I'd like to call the next presenter here. And indeed he is a very good presenter on zebra fish, Mr. Rahul Kushwaha. Are you alone, Rahul?
I'm never alone actually.
Where are your coordinators?
Yeah, zebrafish group. A lot of fishes. I think you people are nothing, right? You need to step up. Do I need to step up? I don't need to, right?
Now we are
Where do you want
moving.
me to be?
from art from to pieces, zebra piece.
No, only I will hide it. No one will hide except me. Why are you hiding? You cannot hide it. Go ahead, go ahead. Go ahead. Only I can hide like this. Don't ask me questions.
Swag will come in, so camera will capture and people will be able to see.
Yeah, let camera capture them.
Yes. You have got enough time,
They
right?
should be captured.
So I am Ekta. I am coming from Kozhikode, Kerala. I completed my MSc in botany.
Speak a little louder and closer to me.
I'm Sonal from
Mic is off. The mic is on.
Dalsing College, Delhi.
I am Dhanraj from Cube Elvinson and I'm working as the Junior Research Fellow for Cube.
I'm Shubhika. I'm from Cube Palakkad. Good morning, everyone. This is Swaha from Cube Jalpaiguri. I'm a 12th standard student.
Hello everyone, I am Rahul. I am from Cube basically, mostly all the centers. And it's good to be here on the stage. And please roast me less. I've been roasted for a decade, so have mercy. We yet have to establish what we are doing.
Okay, so let's start. So here our model system is zebra fish. So we all may familiar with the fishes in aquarium and all. So the fish we are talking about is our system or the Indian model system, but that was invented or discovered by an American researcher. So we want to don't give that much credit in coming years. We should do something using this zebra fish. So we have a model system that is discovered by an American scientist, but later we want to do so many things about this zebra fish. So you all want to join with us in the board presentation as well as the coming experiments on this zebra fish. So why zebra fish? As I already mentioned, this zebra fish is a Indian model system. So that an American scientist who came here and discovered this model system, or he's introduced his model system to us. So here in this zebra fish, if we look around, we can find in our pond. Now we are also getting this zebra fish from our aquarium shop itself. There are different colors of zebra fish. So why zebra fish? So mainly our focus is like we know that humans taking almost 9 months in their development of embryonic stage. But coming to this fish, they are taking only 1 day to reach here. So they are male and female zebra fishes are there. They are laying egg in the water and they are forming zygote and the whole embryonic stage happening within 1 day. So these can be a good model system for studying the embryonic development. Not only that, if we can also visible by our eyes. So it is optically very transparent. We can also use visualize these eggs, the development of eggs in a very visualized manner. So that also make this model system very new or very to make them special about to us. And also, as I mentioned, it is taking only 1 day to complete this much of stage. So the rapid development, we can also say the one of the quality of this model system. And also, it not need more cost for maintaining and all. So these are the things we are very like to be a part of this zebrafish model system, and that's why we are introducing this cheaply available model system to all of us.
चेक किया है या फिर
Wow.
So these are the stages of zebrafish. I am Sonal from Dalsing College, Delhi. So these are the stages of zebrafish life cycle. And these are the pictures collected by all over the country collaborators. We can see the first picture of collection of egg, and the second picture is of fertilized egg, and third picture of hatching. Hatching was done in one day, and then there is a picture of hatching the microscopic view, as Tita was saying. The layer was very clear, so we can see the developmental stages very clearly. And the next stage is fry stage that we can see after 1 month. And we can differentiate between the fertilized egg and unfertilized egg by seeing that they are clear or not. If it is unfertilized, then it is opaque in appearance, and they can become blackish after some time. So Lahiri was joining us
It's online.
online.
Can anyone of us type message?
Yeah, she's online.
Ready?
Maybe chat online.
So we have Lahiri from Nellore, Andhra Pradesh. So she is joined with us. She is a BSc student.
She's there. Just
Hello
say yes
Lahiri,
or no.
can you hear us? Hello Lahiri.
Yeah, Lahari is audible. She is speaking.
Yes, Lahari, you are audible, but voice is not.
Huh. Did you type now? I think she's also facing some network issues.
Okay, I think we can continue on.
The link that you have given is YouTube link. This link
We are facing a little technical difficulties here.
Yes.
So I'd like to ask you to continue.
See audible. Lahari, please speak your audible. Lahari, yeah.
Yeah, this is Lahari from Nellore DW College. This is our observations done by our previous seniors of our college about the developmental stages of zebra fishes, and they have maintained the generations for their further study on fishes. Yeah, coming to next slide. Yes, why this zebrafish is a model organism that we have chosen means it has more special things that the zebrafish have. And as an vertebrate, the zebrafish has a similar and major organs and tissues as in humans. And their muscles, blood, kidney, even their eyes all share many features with human systems. And the zebrafish has a unique ability also. Means it can repair their heart. If it gets any injury or any repair, if it gets any injury, it will repair themselves, means regeneration. And the heart muscles near the damaged area lose their muscle properties and revert back to stem cells, which can repair the heart tissue. For example, if some part of the heart is removed, they can grow it back in a matter of weeks. Coming to next slide. Yeah. I have started these zebrafish model observation on Jan 2023. I have collected 5 male zebrafishes and 5 female zebrafishes from Zoology Department of PS University in Nellore and natural environment to them. So, and also attached the aerators to give oxygen to them. How to identify male and female zebrafishes. Usually the female zebrafishes are a little bit larger than the male zebrafishes. And the male zebrafishes are slender. The female zebrafishes have the larger belly. We can see it clearly. Their abdomen region is belly type. And also the male is like flat. Means their abdomen is not bulged and it is not in a belly type and it is like flat. Coming to next slide. I, yeah, for every aquatic organism, feed is a must and should. We must be aware of which food, which feed we are giving to them. Yeah, we have sugar artemia for our zebra fishes. Means it has an more protein content in it. So it is more nutrition food for our zebra fishes. So we have sugar artemia. It was a saltwater living organism. First, for that, we have taken a 1000 ml beaker and an artemia egg, added to that a pinch of artemia seeds we have added to that, and then added a spoon of salt to it and waited for an egg. Means next day morning, the artemia larvae will be hatched. So after next day morning, we have filtered that artemia larvae with a fresh water. Why we have filtered with fresh water means the artemia is a saltwater living organism, but our zebrafishes are freshwater living organism. So we must feed them by filtering with a fresh water by using a net. By using a net, we have filtered that artemia larvae. Then we have fed it for our zebrafishes. So this procedure will be repeated daily. This was the link before slide. Yeah. There was a link that shows the artemia larvae after it was hatched.
सर क्यूब ज्वाइन कर बहुत हो गया आईटी
Is it not the downloaded video? No? Okay. So, Larry, please go ahead with the next slide.
Yeah, okay, ma'am. Yeah, this was the breeding setup we have arranged to trap the eggs of the zebra fishes for our further developmental stages, especially the developmental stages. We must collect the eggs. For that, we need a breeding setup. So we have collected the egg by arranging one of the traps by using medium-sized glass tray, and we have closed that medium-sized glass tray with a net and a rubber band tightly. And we have kept that setup into the our zebrafishes bed and zebrafishes tank bottom of the middle of the tank at bottom. We have waited for a few days to collect the eggs. After a few days, we have taken it out from the tank and we have observed and transferred eggs of our zebrafishes. So after we collected the transparent eggs of zebrafishes to study and developmental stages, we have observed the egg under the microscope. Then these to start an observation of different stages, we have cultured them in a tray. First, the eggs were collected in a glass tray and cultured it without giving food starting, and it gets to 2 weeks. When the size was increased, we have arranged it to next a medium-sized baby fish tank. We can see it in next slide. The baby fish tank. Yeah, it was the baby fish, zebrafishes tank to know the stages of the zebrafishes. The size was increased from 2 weeks. We have observed the change in their size, and after we have observed, and the next coming to before slide. Before slide. Yeah, the 2 month baby zebrafishes. At 2 months zebra fishes, we have given Atimia as a feed. Before we have feeded it and powder feed means in humans for our babies we will feed an nectar type food means a powder food because they are sensitive to that much of complicated food. So, for babies, we will give a powder form of food. Likewise, our zebra fishes also, baby zebra fishes will eat a powder type of food at 2 weeks and 4 weeks of stage. Then, after it attains to 2 months, then we have given actinia feed for our zebra fishes. Actually, the zebra fishes with genes are 70% similar to the humans. So, by studying of these zebra fish, we can treat many of the diseases caused to the humans. So, if humans get any disease, then we can use these zebra fishes for our research purpose to test and on them for quick result. This was the video link.
Can we skip the video?
Video link, we can see the 2 months baby zebra fishes.
Lahari, we are playing the video. Meanwhile, we can continue with the next slides.
Oh, yes. Coming to next slide. Next slide. These were the stages we have observed after arranging the breeding setup into the aquarium. And these were the transparent eggs. The dark picture shows the transparent eggs that we have observed. And this was the fourth picture that shows the zebrafish embryo under the microscope. Yeah. After 2 weeks, this picture shows after 2 weeks our zebrafish's size. And after 4 weeks our zebrafish's size is, yeah. After a month the size was increased. In the seventh picture you can see that. After 2 months our zebrafishes get a medium size.
Okay, so basically, our aim is to study the innate immunity in the zebrafish. So everybody knows what is innate immunity, is it? So innate immunity is basically when we are born, our body develops a mechanism or resistance to fight the disease. So that's what the innate immunity means. So here we are studying the innate immunity in zebrafish embryo. So why zebrafish embryo? So the embryo has the rapid developmental stages, and as it is at the initial stages, so we are studying the innate immunity in the embryo. So our experimental design is to collect the eggs from the breeding setup and then making test and control as we do for every experiment with the replicates, and then inducing infections in the test setup. So what kind of infections? So many infections that can or bacteria that can infect the fish, or lactobacillus, lactobacillus, or E. coli we can say. then observe and we can do a comparative analysis on the basis of the control.
I think all of us used all our enthusiasms yesterday. So we're just very, very sleepy at the moment, but this should not be the case. Okay. So basically why we are talking about immunity here is, we were all talking about development. So zebrafish is known for being a very good model system to study development because of what Tirtha explained of its optical clarity. You can see each and every cell in the real time dividing, and inside the cell also everything is visible. And what labs with sophistications do is they tag certain proteins with GFP, RFP, and you will be able to see these proteins live under the microscope. So no layer is there which is shielding any information. But then they came up with the idea that why just limit zebrafish to developmental biology? When we were discussing, they were more enthusiastic than they are presenting, maybe because the enthusiasm is a feedback thing. So I can see audience also is not enthusiastic knowing about zebrafish. Okay, they are the other kind of fish, and we have lots of fishes around. But zebrafish are actually very special for the reason that you cannot just ask one type of question in it. You can ask a plethora of questions. Reason that all invertebrates only have innate immunity, as we read in textbooks, and we say that innate immunity is what we are born with, which means like Drosophila, for example, just have innate immunity and it's born with it. So throughout the life, according to textbook knowledge, that's where we included this to uncover the syllabus. Textbook says that you cannot make changes in innate immunity because it's a fixed generalized immunity given to organisms, and over time we see that even organisms that just have innate immunity show responses which vary so the strength of the response vary with subsequent infections. But that is not what fits under the classical model of innate particular virus, right? But organisms do that even just with innate immunity. So there is something at play that we don't know. Evolution can't be so dumb that okay, I'm giving you this, go ahead. Organisms always keep evolving, and even innate immunity is evolving. So the definitions also needs to be revisited. That's where zebrafish becomes a very interesting model system, because embryos mostly are very prone to infections because they are the most fragile, vulnerable part of any organism's life. You are an embryo, any infection happens and you can die because you don't have fully functional organs, systems, etc. But zebrafish embryos somehow are more robust and they can go through infections that other organisms, even vertebrate embryos, cannot handle. So that's where the idea came: can we develop zebrafish by just using bacteria that are not pathogenic to human but might be pathogenic to a fish? So we can just induce the infection and see if the zebrafish still survives and survives normally, was what his idea was. So I think you would like to do some justice to it. Otherwise, no one cares about culturing zebrafish. And remember, whenever we stand on this stage, if I ask you now, which is the best model system? Exactly. So you have to sell zebrafish. No one is buying it at the moment, right? People are more interested in frying it. They're delicious. They're Indian. But we are saying that, okay, don't fry them, culture them. So why should they culture them? Yeah, go ahead.
So, and the zebrafish has the body's ability to recognize pathogens. So, as it shares 70% genome with the humans, so it has that ability.
Also, in the beginning, we missed the chance to say that the only other vertebrate system in cube is zebrafish, right? So yesterday we were talking about dogs with so much of enthusiasm because they are so close to us. If we go a little far distant away, zebrafish are the second closest organism to us in evolution, right? So they are vertebrates, and the good thing is they don't need that much space that dog needs. right? But I think Ketha, we are missing these points. What do you think?
Yes, sir.
Next is the phagocytosis and the inflammatory responses. The infections which are induced in the zebrafish, so that can cause an inflammatory response. And phagocytosis, as we all know about the cell, foreign cells surround the cell and
The slide, previous one.
the other is the components of WBC. So, like we can see the macrophage, neutrophils, NK cells, all are the types of WBC. So, macrophage is basically what prevents the infection of the cell. What neutrophil does, it kills the cell. And NK cell, as we all know, natural killer cells. So it infects and surrounds the cell and kills the cells, as we can see the about cancer cells which multiply and divide.
Okay, so I have one thing to add. The reason I'm standing here to get roasted is the reason I'm putting my life online because Aruna is sitting there back. Where I think he's preparing his set of questions for me probably. Can we go back to the picture where the cubists have very beautifully captured different stages of Drosophila? And I have something to propose here. Sorry, zebrafish. You know that happens when you are in love with one model system.
This is one.
No, the one. Let's go back to the beginning. And I have a few things to propose to people who are willing to take up this model system. Yes, this one. So this is a very important picture here showing the microscopic view of a zebrafish hatchling. What you see in this, so anyone is curious? So the stage is also open for questions. So please, we can also adjust questions in this session if you want to ask. Anyone is curious? What is this? Or we just don't get because what is it? Come again. At the worst, you can be just wrong, right? And it's what is it? They have not formed abdomen yet. It looks like if these are two eyes, then the most natural thing is that it has also not formed swim bladder as of yet. It's a hatchling. It just hatched. It has a very big pouch that looks like a pot belly of some landlord, but it's not that. Yes, but it's something connected. Someone from that side whispered a word. Yolk, yes, correct. So I think we are whispering, we are not shouting it loud. We should shout it loud. It's yolk, right? So zebrafish has a large amount of yolk. When it keeps developing, the yolk keeps diminishing. One of the idea is to check. So this yolk provides for all the energy required by this zebrafish because what people have shown here beautifully is, so you see that there is something, an outer membrane and there is something inside it, right? You see in these fertilized eggs. So this is the actual embryo developing and this is the chorion, the layer in which the embryo is present. So everything that the embryo needs before it hatches and even after it hatches for some time is provided by this yolk. And this yolk provides energy for development normally, but what happens, a good thing to check will be if we keep these hatchlings in two separate, let's say, cups, one with a heavy load of bacteria like E. coli, and the other which has clean water and no bacterial load or even like we can test fungal load. So will it start affecting the embryo because the bacteria is also in that culture, the fungus is also in the culture. You can also put a drop of kombucha tea that we were discussing last time. We can try all these things and see if I put an extra burden on the immune system, which is still also developing. It's not developed in these embryos, but their innate immune system is robust, right? So a straightforward experiment that we all can do is, if we keep these into two separate places, one has a bacterial or a fungal load, the other has clean water. What will happen to the usage rate of the yolk? Now the yolk will rapidly diminish because there are two burdens on the embryo now. One to develop, another to, along with developing, also to keep fighting any possible pathogen that's trying to infect it. So it's fighting, its immune response is taking more energy, plus this is itself is taking more energy to develop. So this was one thing that we have not done. We planned it, but we could not do it because people could not culture a lot of these hatchlings. But it's not very difficult, right? So and we get zebrafish, like Tirtha said, everywhere. Even in like aquariums, these are very, very docile fish. They will not harm you. Yeah. Say. So this is one thing that I propose. Whoever who wants to do this, please do this. And I'll try myself to buy some zebrafish or get some zebrafish and try to encourage, because this is just a very simple idea we can just test, right? What happens to the immune system and to development side by side by infecting an infection. The other thing that's interesting is zebrafish actually show cannibalism. Do you know that? So even the mothers or the fathers both, because there's external fertilization. So how they fertilize, cubists had a real hard time getting eggs, because first we also learned what in this, what the Nellore group managed to do very successfully is, you need to put the male and the female zebrafish for overnight together in a smaller tank where the male can continuously keep chasing and biting the female zebrafish around this fin, and this is basically to bring the female into that reproductive phase, like where she is in the heat period, we call it. And then when the first light is on, female lays a lot of eggs, male always swim just close to female, and the moment female releases eggs, male releases all the sperms and fertilization happens external. Now this whole process is so toll taking and physically exhausting to both the male and the female that they may even eat all their eggs in the same vessel. After they laid the eggs got fertilized or not fertilized, they will eat all the eggs to just regain the energy they lost throughout the night. But don't you think it's very counterintuitive in evolution? You are laying eggs, you are fertilizing eggs.
Then, if they don't themselves survive,
Yeah.
so they won't be able to lay eggs anymore. But if they are able to survive, they can lay eggs again.
But they will eat the eggs again.
I mean, because in nature there is not just one couple of zebrafish, there is a lot of couple of zebrafish.
Right. But they will all eat their eggs. It's not like only one couple is eating their eggs.
But some will survive.
Some
How?
will survive. Because they are settling down.
Jay, please use mic.
Yeah, right. Because this discussion, yeah.
Some of them will still escape because they are in the flow
Eggs
of
can't
the-
escape, right?
They are in the stream, na? So
No, they lay eggs in shallow water.
shallow water still
Which
they
is-
will be settling down. So they may not be eating every egg.
Right, that's one thing that what Jay is saying is the only answer that we have, that wherever they lay eggs, there are shallow water, and because of shallow water, the flow is less. So along with eggs, the pebbles also do not move, they are there. So some eggs go into the crooks and corners of the pebbles in the shallow water and they survive. So is it just survival by chance, because eggs cannot defend themselves in evolution? So this is survival by chance. Some survive, then they become parents, again lay a lot of eggs, eat a lot of eggs, still again some survive. So in evolution how it works is, because where they lay eggs, if you make them lay eggs in this kind of a container where there is nothing to settle, they will eat all their eggs. And you will never be able to understand why they are doing this. They will also never be able to understand, there is no parental instinct. Some organisms go on different lengths to protect their eggs. Some organisms don't at all care about their eggs. These are also two things we can connect zebrafish to. Like, you must be knowing about octopuses. They have been given the best parents in the nature kind of award by many documentaries, because there are species in octopus where female lays eggs. So when they lay egg, they stick the egg to rocks, and then the female sits there guarding the eggs. She does not even move to have food. She sits there for weeks and months, and by the time just because she's sitting there with all that vibrant colors, no predator will try to come and try to eat the eggs. And when the eggs hatch, by that time the mother is already dead, but still sitting there. So she dies so that the next generation can come. That's also an innate hardwired behavior. They will continue eating the eggs, and they will never understand that the next generation is important. So we can also make evolutionary links in understanding this behavior, right? Once you get these eggs, the next big question is the Nellore group and even Lahiri talked about giving some powder, right, to the babies. We do not give them the cultured artemia, we give them powder. You know what is that powder? That powder is nothing but dried and powdered zebrafish itself. So right from the beginning, we are feeding zebrafish a powdered zebrafish. But what do they eat in nature? What do when they hatch out? If we cannot give them artemia in fresh water, what do they eat? Right? Yeah, planktons, right? So they know how to eat, but when we are conditioning them, right? So we are doing an experimental evolution here. Right from beginning, we are just giving them powder, like flake powder, and they are just eating that. So over time, if you go back and keep these eggs in the wild, like after successive generations, will they lose their ability to figure out what kind of food, or they will change their food preference? Because this is what artificially we are doing them for successive generations. There's a very good video on YouTube you should all look at. So if you take up, so mother birds always come to the nest, and just by instinct the baby birds open their mouth and mother puts something in it. But over time the bird learns how to fend for itself and find food, right? But if you take a baby from the nest and you give the food by a dropper, and then this person just left a worm wriggling on the floor, so this bird was going and opening the mouth in front of the worm, as if the worm will just jump into the mouth. So it does not know that I have to actually kill and eat it. It just is going in front of the worm, opening the mouth. Then the worm is moving away, it's just going back, opening the mouth. It's confused what's happening. So how are these behaviors shaped is also an amazing thing we can test through culturing zebrafish over generations, right? So all these things do not need any sophistication. These are 3 experiments you all can take up. And with this, I'll just stop here. We need collaborators direly. And remember, this is also a vertebrate system. So whatever you address in zebrafish, you can, even in terms of genetics, you can compare it with humans very well. Yeah. Thank you. Thank you. Yes.
Yeah.
So I think
So
it's
they will
over.
take the questions. Yes.
So
Yeah.
there are many questions for our presentations. First time. Is it?
Okay, I have one question on the immunity.
I'll just ask. I'll just answer what ma'am asked once. So she's asking: is there a special permission required to culture zebrafish in institutions? So at a household level, zebrafish are also aquarium fish. So if you're culturing it just to study behavior and you're not changing any genetics, not putting any gene in or putting any gene out, or not making transgenics, you don't need to have permission to just culture and study behavior. And all these experiments do not require any kind of genetic transformation to the organism. So, yeah. Yes, Jay.
Yeah, so I had a question on immunity. That
Yes, immunity.
when you say about immunity, what are you going to actually study? Because like, what are you going to test as a sign of an immunity? Like because the immunity you have shown like lot of things are there, NK cells and lymphocytes, things like that. So is there a way by which those things can be, for example, quantified or looked at the levels of, for example, one lymphocyte when you are treating with the bacteria, the embryo with the bacterial infection, the level of certain lymphocytes or some immune components will be increasing. So have you do you have any plan about it? What will be the experimental design?
Experimental design is basically will follow this about the you talked about the NK and the other cells. What I think is the zebrafish embryo has the rapid embryo development, so they are born with immunity. So
But
as
what
of
Jay is
now,
asking is what will be the readout? So he wants to know, if you do this experiment, what are we going to come back and tell that, see, we did this experiment and this is the, so what is the data? Is what Jay is asking, right, Jay?
so like either if we say any change or infection in zebrafish like inflammatory response, so we can confirm that it is whether infected or not, infection is induced or not.
Can you hear me? So I think Vivas online wants to say something. Vivas, if you can hear me, you can unmute and please contribute.
Yeah. I wanted to say that this is a very good proposition that you
Are you there,
made
Vivas?
for the immunity. Yes, can you hear me?
Yeah, we need to keep a mic, I think.
Hello.
Yes, Vivas, we can hear you.
Yeah, I want to say that this is a very good proposition that you did, that you made for the immunity model, uh, because we in our lab is studying a similar model with the mice.
That's great. That's
Uh,
great. Let's yeah. Please help us to design an experiment here.
yes. So what in mice we are studying is we are studying the effect of maternal infection on the pups.
Bivas,
Like
would you
what is
also
the
like
effect
to
of maternal?
Bivas, would you also like to tell about your lab? So because some of these cubists are inspired by you and would like to, look for similar labs or write to you or write to the PIs. So where are you and what lab are you in?
So I am currently working at Yale University, USA, and I I am working in a lab which is basically a neurogenomics lab. So we study neuroscience in combination of genetics mostly, and our model system is like mouse and a monkey. Uh, we are doing a similar type of study that you suggested, Rahul, with the zebrafish. As you suggested that we can put this zebrafish, like fish in a a surrounding which is like enriched with bacteria, enriched with like a lot of fungus and stuff. We are doing a similar type of study with the mouse system. So what we are doing is we are checking the effect of maternal infection on the embryo. So we are infecting the mom with different type of viruses, and particularly we also did with COVID virus. We are doing a different type of bacterial infection, and we are checking that what is the effect that is happening inside the embryo. It's a similar type of condition as you explained because uh, embryo through placenta, passes through placenta, so the embryo has also the will have the bacterial milieu in the blood itself. So to answer Jay's question is like the basic thing we can look for, like what could be the readout? The readout could be anything activated immune cells, which is very easily we can find out by doing staining or something. That could be an easy readout to see whether the surrounding at which the zebrafish is kept is affecting or not. And to be surprised, like we are also collaborating with one zebrafish lab in at Yale. So it's one of the lab who is also studying the effect of maternal infection in zebrafish. So it's a similar type of effect, and they are studying the effect of maternal infection on the embryos of the on the eggs of the zebrafish before they're hatched. So they are infecting the zebrafish with different viruses, and they're checking is there any effect on the embryo itself. So it's a very
So Bivas,
which can
welcome on
promote
board. Thanks for being the first collaborator from this meeting.
collaboration across the model system.
Across the borders.
Across the borders, yes. So Bivas, also I wanted to ask, do you people also look at, can we also take morphological readouts? For example, the infected one, which has a more load on it, the load to develop and also the burden to fight pathogens. So there will be also developmental defects or developmental delays, I should say, that you people also see in mouse and zebrafish embryos. Can this be one readout, I think?
Yes, so developmental delays is being reported in the infected embryos of the zebrafish that we are collaborating with the person. So yes, so that delay we can account for, like how many days it is taking to develop. So that is the
The
also
length,
possible readout.
the length of the embryo, the size, etc. Right? That could be one thing we can begin with that cube.
Thanks, Vivas, for
Exactly,
your
exactly,
collaboration from
exactly.
USA. I'm interacting with you all now. And thank you all guys for that nice presentation over here. This is our second presentation today. And I'd like to invite all the cubists present here to join the next session. So we have to cheer up for the next sessions. So I ask you all to invite to join the next session as a chai session. Yeah. So this is the 10 minutes tea breaks now. We have now 10 minutes tea breaks, and after 10 minutes we'll get in here and start the next session.
Thank you. Suji just opened the that we are going for a interest and come back. Let's just go through the slides. Next is silent. I think so. Supriya, do you know?
Master, no, I guess the beginning energy is not enough for this presentation. Let's make use of it for next 2024.
Let's do these things. Right?
Yes.
I'll just charge my laptop and then I'll be able to work.
Ma'am, is it file and this after this, presentation is Abhijit,
खाली उसका बेचारे का फाइल अंदर का वीडियो देखिए कल जैसा ना हो वापस 57 अच्छा मैम लैपटॉप देख कर मैं पूछना ही नहीं मैं खुद कल्चर कर रहा हूँ भाई मेरे को गालियाँ पड़ेंगी बाद में
थोड़ा वॉइस इधर तो 100% है।
अच्छा
स्मृति मुझे एक बार कॉल रिसीव करने के लिए करेगा। इसके बाद कोई बाहर नहीं आता है।
हाँ आएगा बट इसमें से नहीं आ रहा था 160 क्रॉस नहीं करेगा
Unmute and speak.
समझ नहीं आ रहा
नहीं नहीं जो ऑडियो का है यहाँ पर
हाँ
नहीं आ रहा है तुम भी जाते हो 1 2 3 लाइन
21 से लिए फिर वो लॉन्ग बुक
अनम्यूट करो बाहर जाओ और 2 3 लाइनें बोलो
तो आपको मेरा आवाज नहीं आ रहा है, मुझे आपका आवाज आ रहा है।
वो लास्ट को सुन लास्ट को आपने को भी फोटोज चाहिए ना इसलिए साक्षी का ऐड नहीं किया ना तो फिर कौन हो जाएंगे उसका ढूंढना पड़ेगा तीनों पहले हेडर में दबा दीजिए
I don't know, तुम्हें ना हंसी आ रही है। क्या? हेलो 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 हेलो। 5 मिनट। हेलो 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 what romantic picture like this thing
अभी जब भी वो बोल रहे थे स्मृति कैन यू स्पीक वंस अगेन स्मृति कैन यू अनम्यूट एंड स्पीक वंस अगेन
yeah okay so acha I was muted okay hi hello listen to me we see this can you hear me I'm audible अरे हटो हटो हटो
Okay, Smriti,
am I
can you hear
audible
me? Please mute your mic.
yeah you're what I don't know if you can hear me मैं ऐसे चिल्ला के बोलती हूँ ना
Please come back to the
मैम
mic.
आप hi hello listen to me we see this can you
बैकग्राउंड की चीजें बंद करिए तभी ज्यादा लैग हो रहा है और एक और हमारा पहली खोल लिया है पीछे हमारा इसके बाद वाला है ना
अंदर आ जाओ
Smriti, please come back. Next is mosquitoes.
नहीं इसकी इस वाले के बाद है मस्कीटो फोर्थ है
तो
मस्कीटो
एक्चुअली अगर ज़्यादा फुल नहीं होता है तो स्टार्ट विदाउट ज़्यादा नहीं
तो मैंने व्हाट्सएप आपकी वो स्लाइड शेयर वाली लिंक भी भेज दी
मेरी आवाज आ रही थी
तो एक बार आप खोल के देखिए जो भेजा हुआ उससे स्लाइड शेयर हो रहा है स्लाइड शेयर का ऑप्शन आ जाता है
करना
देखो ये पहले से पूरा है फिर भी ये लोग
हाँ क्योंकि मीटिंग के वक्त ऐसी चीज हो जाती है तो सौम्या विद्या यार तो आप बंद कर सकती हैं 3 घंटे का है उसका
इंटरनेट कनेक्शन वो बंद करेंगे तो इंटरनेट नहीं चलेगा
डायरेक्टली बंद कर
अभी नेक्स्ट प्रेजेंटेशन क्या है कुछ मुझे मिलेगा राइटअप में अच्छा नेक्स्ट
कैलेंडर के बाद मस्कीटोज है
के बाद 11 what
54 इस प्रेजेंट वाला एक बार ऑप्शन दिखा दीजिए वरना आपको लिंक शायद चेंज करनी पड़ेगा
ये मत करवा मैं फाइनल
और कोई बाकी है चाय पीने वाला?
हाँ भैया मैं
इनको चाय, एक चाय ले यार। माउस यूज अरे बाप रे, हैंग कर रहा है। ये मैं वो मॉस्किटो वाला दिखा दीजिए फुल में। ये बेटे के जैसे हाँ एक मिनट। दोनों तरफ स्ट्रीम हो रहा है ना, यूट्यूब पे भी हो रहा है, फिर इधर मीट भी चल रही है। या तो एक लैपटॉप से मीट कर लेते हैं, एक से आप स्ट्रीम करवा लेते हैं। वो पता अभी सब टेम्पलेट वाला सब बंद कर दिया है और नेट ऐसा है तो यार बना ऐसा लिंक्स को आपको नोटपैड में रख लेना चाहिए, तो वहीं से कॉपी पेस्ट कर लेते हैं, ऐसे वो शेड्यूल वाला भी खुलता है आपका बार बार लंबा। नेक्स्ट प्रेजेंट ऑप्शन भी थी ना फाइनेंशियल सारा क्या? अरे वो देख रहे हैं, पीपीटी देख रहे हैं।
शांता सर Philanthus, then mosquito. Philanthus and then mosquito. Okay. Let's just
एक के बाद एक हमारा अगर ये प्रेजेंटेशन ये सब आ भी जाएगा तो उनका वाला लेना बिकॉज़ वो लोग टाइम टेबल फॉलो करके आ रहे हैं
So why is that? Hi Dad. Okay. Moina mosquito. Just
एक और स्टार्टिंग में थोड़ा स्लाइड्स क्विक क्विक रहेगा क्योंकि स्टोरी टाइप का है तो मैं आपको जल्दी जल्दी बोलूँगा ड्रॉप कर लीजिएगा जल्दी खत्म कर लीजिए हाँ मतलब स्टार्टिंग में हमारा जल्दी है फिर थोड़ा सा कंटेंट आता है अब सब स्पॉइल ना करिए समझ गया चल गया थैंक यू
it will take the same amount of time.
अब एक बार खुल गया ना बस उसको वही देखना था वरना मेरे लिंक आपको बदलवाना
नहीं मैंने
पड़ेगा
खोला
हाँ बस ठीक है थैंक यू सो
Moina first. Now the first presentation will be Moina, then mosquito. Moina. Moina mosquito.
It's after eating water.
मोइना का ग्रुप कौन है राहिल
Ryan. आपको वो भेजा ना मैंने काम This is the result.
प्रेजेंटेशन रेडी थी
मोइना को
ना आगे आगे करके देखो
You can come down now, and then you can follow me. You can just This is the Moina driver.
We want 5 lakhs.
The slides are not there. क्या present करेगा?
मोइना नहीं है the slides are not ready they're saying don't we have Philanthus?
मोइना का प्रेजेंटर रेडी है
Take 2 minutes. I just do 3 slides. अपने को present करो. Refresh करो.
मैम नीचे नीचे बहुत सारे स्लाइड्स एकदम नीचे जाओ
52 से ना
हाँ 52 से स्टार्ट हो रहा है हाँ है अब बस नीचे जाओ ना नीचे जाओ
बहुत टाइम लगेगा ना उसको करने में बहन
अरे बहुत ऊपर के हाँ 64th slide मुझे बना था बनाना है।
अरे वो चालू ही नहीं होना उसका लैपटॉप खुल ही नहीं ये बंदे ने ले लिया लैपटॉप
अभी प्रेजेंटेशन इज नॉट रेडी
कर दो ना मैम डिलीट फटाफट हो जाएगा कर दो उसका खुल नहीं रहा था इसमें हट गया
Take 5 minutes. Some slides हैं.
हाँ
There are 60 slides in the morning presentation.
मेरे को है ना आज
Blank है,
कैसे
ma'am.
मेरे से हुआ वो चीज
ब्लैंक है
It is blank house को delete करो।
ब्लैंक स्लाइड्स है
मेरे से मैं ना इंटरव्यू में बैठी थी
मतलब ऐसे
और अभी जहाँ क्या मेरे को बाहर जाना था मैं लैपटॉप खाया और सब
हाँ,
फ्रेंड याद हो
भाई
गया
ले।
ऐसा हुआ शायद
अच्छा that
ऐसा कल रात को थे तो फिर मैंने नहीं किया फिर मैंने नहीं किया
These are all blank slides.
हाँ तो मैं वही बोल रहा हूँ 20 स्लाइड किया सी
स्लाइड बहुत कम है तो वो एक्स्ट्रा गलती से पता नहीं कौन सा
Second भी blank है.
वो मैंने नहीं
हाँ,
किया
सब blank
एडिट
है।
कोई बात नहीं डिलीट कर दो आप
कैसे करते हैं
करना डायरेक्टली
ये टाइम लेगा इसलिए करना
बट इसमें स्ट्रीम हो रहा है ना तो वो मोबाइल
लाइक टू वन टू
तेरे नाती से करना
मैं आया था वहाँ हेड
और आए कब तक हो तुम
हाँ तो मेजर
then we will take more
so what time
around 1 hour maybe
Okay, I can wait for upload होना चाहिए ना.
you are here till 0 o'clock so we first take mosquito in the meantime we can prepare ourselves then we take
Prepare उसमें फिर उसको वापस कर दो.
नहीं there's some mistake in slide so we will rectify that and then you
Okay, so together like
yeah you try
उसका लॉक नहीं खुला था तो उसने डाला
calendars group is the next oh sorry mosquito group is the next so Santa sir let's take
Mobile. The mobile.
what is the next presentation that we are taking mosquito then fireflies so you can call these people this is
राहिल है
ये टीम
इनर टीम लेते हैं
ये सर
राहिल ईशा सज्जन राहिल ईशा
Welcome back after having a cup of tea, and we are going to start the next sections. We are going to start the next session with a little organism, smaller organisms that fly here from Kanpur. I'd like to invite Ina ma'am, Rahil, Isha, and Divanjali to show us guys. And please make sure you finish your presentation within 10 minutes.
let's clap for them
Let's hear up.
yes sir
then the cycle continues, and our female lays eggs, and the cycle continues. And next slide, please. Now, Kavanjali will tell us about how to.
Next slide. So I'll be discussing the data collected from the claptrap method. So basically we were three collectors, Hina ma'am and Rahil together, me and Kavyanjali. Hina ma'am and Rahil's location was CLN Mall, and they identified 18 Aedes mosquitoes and 5 non-Aedes mosquitoes. My locality was Swaroop Nagar, and I identified 10 Aedes mosquitoes.
Can you go without the slide for 2 minutes? It's a data.
And 1 non-Aedes mosquito. In Kavyanjali's locality, which was Halsi Road, she found 6 Aedes mosquitoes and 2 non-Aedes mosquitoes. So this graph represents the same. The dark blue color represents the Aedes mosquitoes and light blue represents the non-Aedes mosquitoes. Next slide. So now Kavyanjali will explain the OV trap method.
So we set up OV traps to identify the breeding of mosquitoes, as mosquito usually lays eggs in these OV traps. So for setting up an OV trap, we do not need to buy anything. We can set up OV traps from materials we find. First of all, we need to take a plastic bottle and cut it in half, or we can also use any plastic container which we have at home. So after cutting the plastic bottle in half, we use a plastic scale or a bark of tree or even wooden ice cream sticks,
and cover it with white cloth. We use this as base, and to tighten it up, we use rubber bands or threads. Then we make 3 of these OB traps with 3 different kinds of water: muddy water, tap water, and drinking water.
Breathing in the mic.
Muddy water, tap water, and drinking water. And this is how we check the breeding of mosquitoes through OB traps. Next slide, please. So now I'll be discussing the data collected from my OB traps. So I set up 2 rounds of OB traps. Round 1, which I set up from 9 June to 23rd June. I took 3 different types of water: muddy water, drinking water, and tap water. In my muddy water, eggs were seen on 19 June 2023 and its location was in the garden. In my drinking water, I did not observe any eggs and its location was on a windowsill. In my tap water, I saw eggs on 16 June 2023 and its location was the storeroom. In my round 2, I set it up from 24 June to 8 July and I did not observe any eggs in any of my OB traps and their locations were over drain, living room and under ACs. Next. So these are the visuals of my OB traps as you can see. Next slide. Next slide.
Previous slide.
Data.
I'm really sorry for the.
So round 1 OB traps were set up from 9 June to 23rd June in 3 different kinds of water: muddy water, drinking water and tap water. Muddy water was kept in a balcony, drinking water OB trap was kept in a storeroom and tap water OB trap was kept in garden under shade. I found no eggs in any of the OB traps. Round 2 OB traps were set up from 24 June to 8 July in 3 different types of water again: muddy water, drinking water, and tap water. In muddy water near windowsill, drinking water and the previous slide. Drinking water near plants, and tap water under AC. Again, I saw no eggs in any of the OB traps. This is the visual representation of the same data. This right here is tap water, this is drinking water OB trap, and that is muddy water.
Now I will tell you about the prevention methods against mosquitoes. First, we can use an effective repellent like a head or a lotion that we can apply to our body. And the second is that we can stay indoors during the peak biting hours, that is the dusk and the dawn, as Aedes aegypti mosquitoes are commonly found active during those hours. Now, Kevin, you will continue.
So the third preventive measure we can take is we can use mosquito nets to prevent mosquitoes from entering in our homes. So we can use mosquito nets on the windows as well as over our beds. And the second preventive measure we can use is to pour out stagnant water. Whenever we see water collected in an area after heavy rainfalls, so we should take out the water. And these are the orientation photos of our Cube National Meet 2023. This was when we explained about our mosquito model organism, and this photo when Rahul sir explained us more about mosquitoes. So now I'll be talking about goof ups, celebration of goof ups. So I had a major goof up in my round one OE trap. I used cardboard as a base for this round one OE trap, and eventually in 2 to 3 days, the cardboard sunk to the bottom. But what an interesting thing it is, I still found eggs in this OB trap. What could have happened was the mosquito could have laid eggs on the cardboard and the eggs sunk along with the cardboard into the water. Now Kalanji will tell us about her.
So this is one of my the same. This is one of my OB traps of muddy water, round one. This was a goof up I observed after heavy rainfalls in Kanpur. I observed many insects in my OB traps, due to which I had to remove the OB traps. Which, the insects were usually the ones we see after rainfalls. And this is the second goof up. Instead of mosquito eggs, I observed algae growth after 6 to 7 days. And this photo is of round 2 muddy water OB trap.
Now, am I audible?
Yes.
Actually, the mic was off, right? So now after the entire thing, how can we integrate mosquito? This is actually the most important thing. This is a small flow of our research. Of course, we have got good clues from Rahul this time, and we are going to take up questions like olfaction. We have got research links which he has shared. And learning is very important here. Mosquito, how can we integrate, how can we show in a map that it is related to different kinds of subjects, If you see mosquito, we can just study mosquito association. If you are studying rotifers, bacteria, yesterday I was just seeing the microbial discussion, and I just shared to the student or the teacher that microbes may Escherichia coli pe the larva feeds a lot. So there is an interaction here, protozoan, nematodes and algae. We can always study mosquito in collaboration, and the subjects can be integrated. We can study the frequency, the microevolutionary study, genetic variation, adaptation, evolutionary biology and inheritance pattern also. Now, it is so important to integrate a model organism with different elements here. Different Nobel Prizes have been given to this mosquito. It's a very small insect which has not been exposed to the study. 1902 Ronald Ross, 1907 Alphonse Lever, 1948 Paul Miller, and 2015 Yu. So these all Nobel Prizes have gone to this little fly. And still we are thinking it's a small fly just flown from Kanpur. No, the studies have been going on since many years, and there is a lot which we can do, and we'll welcome collaborators. We had our collaborator Shankar, Gorangi, Abhijit has given a lot of data. I thank you all from the deepest core of my heart. And I would want all of you to please give in data and collaborate. Then we can study ecology, control measures and diseases. With this, I would like to thank you all. For any questions, the students will take up.
Thank you for nice presentations. I wish we could have a big applause. This is not only the claims we have given them. This is claim trade for the mosquitoes. Now,
Which slide?
you may ask questions. Yes, Rahul.
So first of all, I think if we had a best presentation slide, I think we all agree that this by far is a very well worked slide. It's very beautiful slides. All of you have worked so hard on the slides, and it's very important to show your data also. So a round of applause for you for making efforts to make this slide. It's really good. And yes, more clap traps. And my question is, so we see these mosquito-borne diseases. They have a pattern which is also related to the geographical and climatic conditions. So like you also said, when Kanpur rained heavily, there were more of these. So normally when you are doing these readings, you would also like to incorporate data like the temperature, time of the day when you are taking that reading. That will also help you to make an activity pattern where we say that dusk and dawn is the most. So are you planning to incorporate that also? Like Arif has done something of that sort in the past. I think that could be one thing you could add to your protocols.
Yeah. More questions?
Congrats, ma'am. Very nice presentation. Kudos to you all. I want to ask you one thing. How could you protect yourself and others while culturing the mosquitoes? Because these are all pathogenic organisms now. So, how?
I think we could use mosquito nets if we want to culture. Otherwise, we were not culturing till now. We just found stagnant water in plant pots, and we saw larvae in them after it.
We use
Yeah, we cover the traps. We cover the traps with thin mosquito nets from which we can see through, but the mosquito cannot escape. The adult mosquito cannot escape it.
Ma'am, actually, we are not culturing mosquitoes. First, this is a very important thing we want to clear because this is a strict instructions from me to the students that we are only giving information of the breeding site where we are finding mosquito larvae and the prevention. Like Khalasi lines is a highly. Kavyajali, you take it.
I think you are using orbiters for the study of how they develop. From eggs to
what kind of
mosquitoes.
eggs are being bred? We are just telling which type of mosquitoes will be there in that area, and then when we are sending messages to the Nagar Nigam. There are sprays happening after every day. Halasi line is being formed every week now, just because of this work.
Now we have one more question from Vivas, I think. Is it Vivas? So the question is, does it is lay eggs on water or near water?
Surface of water.
It's Sarita. Sarita ma'am.
Oh, it is from Sarita. Yeah.
Collaborator. Well, thank you for the question, ma'am. The Aedes mosquito lays on the surface of the water, as far as I know. And
In your trap, where do you see the mosquito eggs? What is that trap? Can you just explain it?
can you go back to the slide of the trap?
That will explain. No, that will tell us the story. So, why are you using a trap and what does it tell us? Please come fast. What is the design of the trap?
Sir, we use plastic bottles. Yeah, we use plastic bottles and we cut it in half. Okay. Then we use a scale or any bark of the tree for holding.
No, tell us one particular thing what you are seeing there. What you are seeing there, why don't you explain to us?
Yeah.
Not all, because that's confusing.
Then
you have
near
the bottom of the bottle cut.
Yeah.
You are using that.
Yeah.
Is that right? That we can see it. Okay.
So they lay egg near the
No. What is that slanting thing that we are seeing?
The slanting thing.
scale covered with white
Please
cloth.
explain that. Please explain this setup.
Yeah.
So we used a plastic bottle, we cut it in half, and this thing right here is we used a plastic scale, this was a 15 centimeter plastic ruler, and we covered it with white cloth.
Why?
To see the eggs, because mosquitoes.
So, where is the egg being laid? That's the question. Sarita was asking.
On the surface of the egg, on the surface of this scale.
So, not surface of the water, surface of the water. He says that it's
on the same level. It's on the same level.
If it's on the surface of the water, why do you keep that scale? Sorry, I'm sorry.
Yeah, even the same question is being asked
So, is
by
it a ritual
Sarita
that you are
ma'am.
keeping a
Follow-up
scale
question.
there with a white paper?
No,
You are keeping white
we are
paper?
keeping it because the Aedes mosquitoes are known to be sticky and they stick to the nearby surface. So if the mosquitoes lays eggs on the surface, it further moves automatically and sticks to the scale, and then we spot it and share the data.
It makes it easier to observe.
Yeah.
Yes.
So the eggs will all move towards the scale.
Yes.
Yeah, they are sticky and they stick to the surface of the scale.
Not on the other side. Don't go to the other side.
It could
No,
go.
this is impossible for us to believe. You are using a ritual of a scale and you are asking the eggs to move towards that scale. See,
This is an experimental
that's
setup.
why Sarita is asking this question from Delhi. And Sarita has been working on it.
Yes.
So she wanted it to be exposed. You are saying on the surface of water, that means it's floating around.
So Rahil, so I think how does the scale help in getting those eggs attached to it? So maybe
Because
if we ask
it is covered by a white cloth and Aedes eggs are generally black in color. So if it sticks to the cloth, we can easily identify the black spots on it.
So do you mean to say the eggs are getting attached randomly
You
just
have a
about
photo.
everywhere?
Yeah.
You have a photo of it. See, the problem with for us is that it's all very
So are the eggs just attaching to the scale or they are getting attached just about everywhere randomly?
Only Aedes mosquito will fly. Sir, actually they lay egg. We have used this scale actually as an OV trap because they come and lay egg near the water surface, near something. There has to be a wall. They don't lay egg on the water. They egg, mostly the Aedes egg, are laid near the surface. They have to have
So
a.
it's very different from what Rahul was telling.
He was not able to explain it, but yes, this has been used because this near here we'll find the egg and of Vyom Gupta, his data he has not actually we have not got his slides here. He has not shared it. Otherwise, in 2000 I think 20, we had spotted white 30 eggs. We have that slide in the last data, and those eggs were visible. And this time we were not seeing eggs on the ruler here, sir, but they were found floating because of the sinking of the cardboard. And we were trying new things because.
What is the logic of this trap that you have designed?
Sir, the logic
That's
of.
the worry of Sarita. I can imagine that.
So the thing is.
If it is floating on water, why do you need a sign stuff like that? Why?
So that the mosquito comes and lay eggs near the
Yes,
surface.
so mosquito is attracted to that. That's all.
So one question. So if we do not use the scale, would the mosquito not be laying eggs on the edge of the container itself?
So this is an experimental setup because edge of the surface we'll try it out.
So maybe I'm just asking in the sense that maybe we don't require the scale. Maybe it's laying eggs on the edge
That
of the
we'll
container
work
itself.
upon. We'll set up an experiment where we'll not put in ruler and this thing, and we can just leave an open container.
And
We can just leave an open container and we can try this experimental setup too. Because this was an open trap setup which we did along with Shankar from Kochi, and we have been replicating it because most of the guidance which we are getting is that try to give them a surface to come and lay egg. Because we are basically working on Aedes aegypti.
so this is the same logic that I'm asking, that the surface would also be at the
The
edge.
wall, yeah, sure,
So that's
sure.
why.
Yeah, we'll do a comparative list. Any more questions?
Yes, ma'am.
No question. No basic.
So there's one more question by Sarita.
Female or female. Identification. The bristles of the mosquito. Female does not have brushy bristles, and the male has brushy bristles. Photo है उसकी एक मिनट मैम स्लाइड आप
Forward or backward? Special purpose.
So here we can see this is a male mosquito. Here you can see the brushy bushes of it. Okay. So here you can see the same mosquito, but it's a female one. It only has a single line-like bristle. Brushy bristle, yeah. This one is the male. This is a female. Yeah. हाँ ओपन ड्रेनेज में अगर आपके स्टैग्नेंट वाटर है जहाँ पे पानी रुका होता है वहाँ पे मूविंग में नहीं हो पाता क्योंकि उन लोगों को सेटल होके देना होता है.
So the mosquitoes they stagnant water.
Yeah, stagnant water. This water which is still.
especially at certain angle or something, or anopheles varieties.
Yeah, Culex egg lays eggs in a raft-like structure. There is a GIF of that. Ma'am, can you please move back? One more slide, I guess. One more.
Are they require any angle setup of something? That's why you're doing that setup, OV trap or something. Are they need any angle or pattern for laying a egg on that surface?
Yes, sometimes because they either sit horizontal, like at 45, the AD sit at 45 degrees and Culex sits on the surface of water. You can see this is a Culex mosquito laying eggs on the surface in a raft-like structure.
That's why you are choosing that OV trap.
Yeah. Yeah.
That is the reason, one reason for that. What will happen if we covered that all the water surface by putting oil on
So the mosquito couldn't lay egg. Even if the larvae are present in it, it will die because the larvae need air to breathe, and they come up on the surface of water and breathe through their siphon. And if you put oil on the water surface, the oil and water does not mix, as we know that. The oil surface won't let the siphon get out and let the mosquito larvae breathe.
So I think this is belong to Culex mosquitoes.
Yeah,
Sorry? And you are talking about Aedes mosquitoes only in this presentation. So why you showing the Culex mosquito laying eggs?
Aedes and non-Aedes.
You are taking as a non-Aedes mosquitoes. You are fine. Okay. The same question as asked by Professor Aruna. It is clarification asked by Sarita from Delhi. If it lacks on water surface, then what is the need of board stripe? Standing board stripe is clarification she has
We are providing the surface to lay egg, so that we can find the contrast of the egg. Otherwise, on the water surface, it's very difficult to detect. Otherwise, on the wall surface also we can do that. But now we'll be trying out without the ruler. As per the questions being raised, there is one more method which we'll follow. Yes, Abhijit.
Yeah, again regarding this life cycle, Sarita also asking, is the species name written in the correct manner? Please check the life cycle.
Oh, egg?
Egg larva, pupa and adult.
Why is it
Arrow is not the arrow. It's not up arrow. It's just a design.
confusion is because adult is coming below
No, it is a adult mosquito emerging from a pupa.
Can you hear me? Okay.
It is the adult mosquito emerging from a pupa.
For explanation, I use this photo.
This is not an adult.
Yeah.
The confusion about the OV trap, which what my idea about it is that when we used to do it when we were in school and we used to discuss with other collaborators is that when you prepare a OV trap and you keep it somewhere, there are 3 types of eggs you will be expecting. One will be laid by Aedes aegypti, and it has a very specific color on the outer layer. I think it will have some spots as well on the egg. So it is very clear to identify Aedes mosquito egg on the slant, and it always requires substratum. So substratum is very specific to Aedes mosquito's egg. But the other eggs which are there on the water, they will be only of either Anopheles or Culex. And how will we identify that? Culex always lays eggs in a group, and Anopheles or mosquitoes lays eggs in an individual pattern. So the purpose of this slant scale is for Aedes mosquito.
Yes.
And that too is very easily identified because it has a specific color on the this
It is sticky in nature. It sticks onto the
Yeah.
wall. It prefers the side wall
Yeah.
or the side. Because we are doing a study and we have to basically Aedes is breeding in our locality, and dengue prevalence was very common. The data, I'll just share the data of the last 2-3 years, even during the coronavirus time, we had the hospitals full of dengue cases. And this is so important for the people to understand, and still the ignorance is there. There are tires with water, there are small containers all over places. Littering is very common in Uttar Pradesh, but the awareness that it's such a dangerous disease where thousands of people died. And especially at the school level, when we are connecting to the curriculum, and there are questions in class 12 CBSE examinations being asked on Aedes aegypti and how the genetic studies will happen in future. They are called, they are talking of control measures. So it is a part of our curriculum. That's why it is of interest to us. But we would welcome all the undergraduates to please come up and join and share data with us. So that we evolve in this model organism and learn more and do a better research work. If any more questions?
Ma'am, I would like to ask a question.
Yes.
It is how many types of water we use? I would like you to repeat once.
They will answer.
We use 3 types of water: muddy water, drinking water, and tap water.
Please show a slide of 3 types of water.
So we know we know that the drinking water, we don't have any breathing there, right? How do we did? We did have that. I
We
will
wanted to
see.
test that only, that's why we just use that.
Can we see this slide first?
Which one is it? Which slide?
Normally, Aedes mosquito breeds in cleaner water, not in dirty water. Can you please give a clarification? Normally, Aedes mosquito breeds in clean water, clean stored water, not in dirty water. But we have found out that the mosquito is breeding in planters. These are the planters in my botanical home lab. This is the baby cube lab in Mall Road, Kanpur. It is my whole little garden in my flat where I trap mosquitoes, I get butterfly pictures, I have philanthus. So this is my, it's a small open lab. You see? Yeah.
So, ma'am, you found out Aedes larvae, Aedes mosquito larvae, and all in your pots. Breeding, they were breeding in the pots.
They are potholes, small plastic planters which are being hung for plants. But when the rainwater is coming down, because of the height of the flat, I'm on the 5th, 6th floor, the rainwater collects there. It's not that we are putting water and breeding. After rain, the 3rd day we spotted larvae in our planters. So we have shared this recent photograph of very continuous. It was raining for 4-5 days. On the 4th or the 5th day, we found small larvae, and he said, now finally the rainwater, we have got larvae. Otherwise, I was continuously getting sprays and all done because naturally we don't want that our study should cause harm to our community. And in our building also there are labels put. I'll show how we are bringing about awareness. We tell people to put at least salt in the water if there is accumulation. That kills the larvae.
Ma'am, can you just, Kiran ma'am, can you just quickly move to the observation slide?
Okay, ma'am.
So here in the distilled water, right? Am I right? So there are no breeding.
It's drinking water.
Drinking water. So in the drinking water, we see that there are no breeding, right? So why do you think that mosquitoes did not breed in the drinking water?
Who said the plants? It was indoor maybe, but not the right locations. Maybe
But ma'am, all the other locations were indoor, right? So
no, this is ma'am. Please see the slide. Garden and storeroom. It's a set of shading.
So basically, the drinking water was set up on a windowsill, which was like kind of outside of, it was in the middle. So maybe the location could have been wrong. And there is also a fact that at that time we had seen rains in Kanpur. What could have happened? The mosquitoes could have gotten better breeding grounds because there was collection of water in potholes and etc.
So, what made you think that, like, you said that the rainwater, they could have got a better breeding ground. So, what made you think that distilled water was not a better?
It's not distilled water. It's drinking water.
Drinking water was not a better.
I'd like to finish the session here, but before finishing the sessions, I'd like to add something from myself. So, as they do the setup here, you see 3 different types of water. They use 3 different types of water for a coffee trap: muddy water, drinking water, and tap water. But they use 3 different locations for the 3 samples. But I would think, for a better result, we must have all the samples, all the types of water of different areas like gardens, all the 3 types of water in the garden, indoor also. So in that way we can have a better conclusion or findings. And one more thing, it is also a clarification. We get the last question from Sarita Kumar. So it is a simple question: can throwing stagnant water help erase prevention?
Yes.
Okay. If the eggs are laid on the surface near water, so you say yes. So we'll have discussions over our Q2 contest group. And one more slide I want to show here. Please go. Can you please go back to the life cycle? No more question. I'm not going to show the life cycle question. We are all doing here.
This is settling a score, not asking questions.
No, we have no time. Time is not permitting us. That's why. Otherwise, we'll have more sessions. So here, we have simple model system to get answers for big questions. So here, you are using the arrows. That makes confusion among us. Because you are showing the arrow here, but putting another dot on the tail side also.
Actually, sir, the idea was that I would explain it like this: the eggs develops into larvae.
Yeah, I know that. We come to know that. But the confusion I'm saying about the confusion, while using the dot, it makes the confusion. So please simplify it next time. Use only the arrow, not any other dots on other places. That's all. And thanks again for the nice presentation. We are going to the next session. No more question, please.
It was meant that it continues.
We'll have that online.
The same person has spoken. Come on.
Yes, please.
Yeah. Yes, again. I'm going to ask the next session.
Thank you.
Please. And please come. People for the presentation of Philanthus. Philanthus group is going to present the next presentation here. Lakshmi, please come up to the dais. The stage. And start the presentation. So please go ahead.
They can introduce themselves.
Please introduce yourself and start.
Next we have Philanthus presentation. So I am Tirtha. I am coming from Kozhikode, Kerala. This is Swaha from Q Jalpaiguri.
This is Sakshi from Q Mandu.
I am Shrutika from Q Homeland, Pattabadalam. आप सभी को जय हिंद. मेरा नाम निधि देवदास है। मैं छत्तीसगढ़ से हूँ। मैं अभी चैतन्य साइंस एंड आर्ट कॉलेज में फाइनल ईयर की स्टूडेंट।
I am Amagaki and from Kerala. I'm just from Cuba.
Aditya from Mumbai.
One important collaborator is sitting there. She's Lakshmi from Kerala.
Okay. Okay. So you all can actually stand a bit closer to the screen so that you will be visible in this camera. Move around.
Divya, please come.
Call all your collaborators. Yeah.
Divya.
Please start your presentation. Whatever you tell your names now. Introduce yourself.
So, myself Lakshmi. So today we are presenting our model system file address. So how we can identify file address? We can see this. We can identify file address by looking at its future, like by seeing some flowers and fruits which are hanging under the leaves. So that why we call it as a file address. So in our local languages, so Pylanthus is a small version of gooseberry. So we are familiar with gooseberry. So it's a small version of that. So we can in Malayalam we call it as Kiras Nelli, this plant. In Hindi we call it as Buyavla. So in the name itself, the name suggesting saying that it's down to the flowers and fruits are hanging down.
Is it the video on the next slide?
So I'm going to talk about Philandus flowering as weed. So we can see this planting everywhere. So it is a monsoon plant, and we can see this plant in various parts of India.
So this is a video depicting the opening and closing of leaves in Phylacus plant by Susanta sir. can see the leaves are opening. So we are trying to see the circadian rhythm or the leaf opening rhythm in this plant. So if we look that video, we can see the leaves are opening. So we are trying to convey the idea that okay, sir. There are also other plants that are showing circadian rhythm, namely oxalis plant, Mimosa pudica, Biophytum sensitivum, Tamarindus indica, and some species of Cisalpinacea family. Tea family plants. Okay, so then Jeffrey C. Hall, Michael Rothbard, Michael Young got Nobel Prize in Physiology or Medicine for identifying the molecular mechanism of circadian rhythm in 2007.
So next, Lahari is here for explaining the solstice. Lahari, are you?
Is Lahari there?
Lahari?
Yeah, she's around.
Lahari, are you around?
She's not coming now. So please go to the next slide. Please come and tell us about this.
Or maybe Lakshmi can explain.
So we have observed there is a phenomenon called solstice. There are four solstices are there. We have summer solstice, which is the longest day. Longest day, which is on June 21st. And also there is winter solstice, which is the shortest day, which is in December 21st. And also there is autumnal equinox and vernal equinox, which is actually the equal day and night. We have equal day and night. So here the Tirtha, Lahari, Abhijit all observed about the autumnal equinox, which is showing here. They observed on September month. They observed the leaf opening and closing of the leaves. They observed how does the sunrise and sunset, which is coinciding the opening and closing of the leaves. So they find observation, they take observations and they found that the opening and closing of the leaves is not exactly coinciding with the sunrise time and sunset time. These are the observations. Likewise, Tirtha, Shrujal, I will take observations. Abhijit. And next is summer solstice. It will explained by Inas.
She's around. You can address her.
Inas, could you explain? Inas?
Is Inas is there? Okay, so as Lakshmi mentioned, here we are now first we observe that the leaf opening rhythm is the yes, Sinas, please say.
Inas, can you speak? Are you speaking?
Yeah.
Okay, so here we are now trying to look about the summer solstice. So as Lakshmi mentioned, we believe that these are the 4 seasons we are getting 2 solstices and 2 equinox. So does the sunrise and sunset time have any role in the leaf opening and leaf closing behavior in Philandus? So we believe that the equinox and the solstice are the two seasons that we can check whether the time have any role. Likewise, in summer, we do have 2 years data, that is in 2021 as well as 2022 data. So here we are representing the summer solstice study that we did in 2022 as well as 2023. So these are the observations we were taken from different parts of the country, including Kerala, Bangalore, Mumbai, Chhattisgarh.
Oh, there are so many people.
Uttar Pradesh. This is the Inas, and also West Bengal and Assam. So we want to take a look into, does the sunrise time and sunset time have any influence in the leaf opening and leaf closing rhythm in Philandus? That's why we have chosen these seasons. We believe that this is the best time to observe the leaf opening and the closing rhythm in Philandus. So we concluded that if the sunrise or sunset time, the time does not influence it. Like the sunrise time or sunset time, if we can take an example like the sunrise time in Kerala is 6:10. But the time the 6:10 a.m. comes, the leaf is not suddenly opening. That is the case in the sunset also. So we have found that there is not such a coinciding in the sunrise and the sunset time in both summer and winter solstice. So other than this time, there is something internally regulating the rhythm in this plant. So from the first video we will see that the leaf opening and closing. From there we will try to think that if the time have any influence. So we conclude, in this observation we are just concluded that time does not have any influence, but we will not stop here. We have coming years also to check whether our hypothesis is true or not.
And also we want to compare with the latitude and longitude, because from Kerala to Assam it is the geography is like different. It's Kerala it is around 10 degree, 10 degree north, and Assam is about 27 degree north. So there will be differences. So Shansar also brings some Philandus plants to test whether any difference in the opening and closing time when comes to different parts. So next we can how we know that the plant is started to close. So at what state we can say that the plant is started to closing, and how it is completely closed. So for checking that, we have developed a new method called leaf angle titration. So here it is showing that this is a single leaf of Philandus. Here we can see the angle we are measuring. Like we do titrations, we try to find out the endpoint. So likewise, we are trying to find out the endpoint of this wakefulness. that is the opening, and when how they get started closing. So here's the single leaf of hyanthus, and we are measuring the angle between the stem and the leaf. Here the A line representing the stem and the B which is representing the midrib of the leaf. So from while opening to till close, we are actually observing how the angle is changing. So so we are taking photos at different time intervals. From the leaves that started to open, it took nearly 1 hour to completely open the leaves. So within that time period, we are actually taking 15 minutes intervals. We are taking photos, and after that, we are taking photos of 1 hour intervals. And after that, on the time of closing, before 1 hour, we take 15 minutes intervals itself. Taking photos, and we are measuring it with an online protractor. So these are the observations taken by Tirtha. So at various time, here you can see that the angle of the leaf is get decreasing. So this is the graph for 3 days, and we plotted it here. So we can see that the angle of the leaf get increasing when the time of leaf opening. After that, the angle is not always constant, even though it is the daytime. It was there was some little bit differences in the angle of the leaves, and when it time to close, the angle of the leaf again decreasing. It is about nearly, if we can say that less than 50 degree, we can say that the plant is in the closing mode. So this is the observations taken by Shaurya. Like here, so he found that in his house, normally the plants that sleep at around 5:26 p.m. But when he observed the phyllanthus plants on the street light, so it is not closing the leaves completely even at 9 p.m. So why is that? So there was the one reference like light pollution, because now in the streets are around there is light everywhere. So even in the night itself, so the light that may influence these plants opening and closing time. So that we need to find out. So I will go to Nidhi.
मैंने फाइलेंथस प्लांट का स्टडी अपने होम लैब में ही किया है। मैंने 2 डिफरेंट स्पीसीज ऑफ फाइलेंथस का कंपैरेटिव स्टडी किया है। फर्स्ट इज फाइलेंथस एमरस एंड सेकंड ग्रीन स्पॉट इज फाइलेंथस यूरिनेरिया। मैंने इसका स्टडी डेट 12 जुलाई 2023 को किया। Evening time 6 to 49 p.m. Next slide. Both phyllanthus plant are leaf open. Then same day and change time 5:30 p.m. Only one phyllanthus urinaria plant leaf are close that time, but not close phyllanthus amarus plant leaf. Next, then time 6:30 p.m. both leaf are close. तो मैंने यह स्टडी किया कि फाइलेंथस एमरस एंड फाइलेंथस यूरिनेरिया में डिफरेंट टाइम में बायोलॉजिकल रिदम लीफ ओपनिंग क्लोज टाइम होता है डिफरेंट।
So now, as she mentioned, she studied 2 different species of Phyllanthus, Phyllanthus amarus and Phyllanthus urinaria. So currently, there is a hot topic going on in this Phyllanthus kind of thing, that there is a controversy in the Phyllanthus species. The controversy is actually, we can find the difference between the literature. Some say that Phyllanthus neruri is a herb which is found in India. But whereas many literatures are available which say that it is actually native to American land and not in the India. So this is the controversy which is going on, and there is differences in the literature which we get whether the Phyllanthus neruri is actually present in India or not.
So there is also speculation that the three species of Phyllanthus, that is Phyllanthus amarus, Phyllanthus fraternus, and Phyllanthus urinaria, are closely related to each other in their appearance also. They have a minor differences. So are these three species together, like a complex, are being what we can say misidentified or misaddressed as Phyllanthus neruri? And it is of the question is of great importance because of two reasons. Because firstly, the taxonomy part and morphological characterization part, which many of the students might not like in their academics, because we simply say that the leaf is serrate margin, blunt end, blunt tip, five petals, everything and everything and everything. But when we give the context of medicinal uses to this morphological characterization, as this Vuiavla or the Phyllanthus is very much important and is a traditional herb which is used in treatment of various illnesses in tribal and local areas like hepatitis or many of the blood diseases or some kind of thing. So generally people make decoction of this Phyllanthus and they drink it. So if the species itself is not identified properly, can it be possible that people here are taking some different species and getting treated and the name is of Phyllanthus neruri, or in place of Phyllanthus neruri they are taking some different species and complaining that no, the Phyllanthus is not working. So this touches upon the great area of taxonomy and placing and identification of the species as well as the medicinal importance which is prevailing in the regions and local level. So, and with the help of this particular question, we can actually create a mass campaign all over the nation. As Phyllanthus can be found in many of the places, why not just look in the environment and just click the photos and just compare? It's quite possible that we may not find Niruri in India, so we may have a reliable data. It's quite possible that we may find Niruri in India also. So let's see, and we hope we can generate a mass campaign of just looking at the Phyllanthus, and by this we can also touch upon the biodiversity in one's own area.
So here I'll be talking about the whitefly interaction. On my quest to identify the Phyllanthus plant I found in my homeland, I saw some unusual growth on the underside of the leaf which looked like a fungus. Or later in the chatShaala discussions, we identified that it is due to an insect or a pest called as whitefly, which is so small, it's less than 2 millimeters in size. So this was the appearance that I found, and we can see that there are some black spots over here, which is nothing but the fungus that is growing on the whitefly excreta, which is the honeydew. And this honeydew is also attracting other ants and minivers. And this was the association I was seeing on the underside of the Phyllanthus. Next slide. We found Phyllanthus, we found whitefly on many different plants. So whitefly is a pest, it's a common pest, and it is found, the cubists found the pest on capsicum, tomato, and different species of whitefly. We found these different species of Phyllanthus. We found the whiteflies on in its various stages, the egg stage, the larval stage, and the adult stage. The one that I found was greenhouse whitefly. Next slide. One interesting fact about whitefly is that on discussion we found out that whitefly was it led us to frontiers of molecular biology. It was one of the very first evidence that suggested horizontal gene transfer between a plant and an insect. Now we've always associated horizontal gene transfer with prokaryotes like bacteria, but what we observed in a paper that was recently published in 2022, that the whitefly carried a gene from the plant and it is using the plant gene to survive on the plant. So plant produces a chemical, a toxic chemical which kills the whitefly. However, the whitefly has acquired a gene from the plant which produces an enzyme to neutralize the toxic chemical. This gene that plant produces was as a self-defense against its own toxic chemical. However, it was seen that the whitefly acquired the gene from the plant It may be viral transduction, that is the white fly and the plant have a common virus that allows the transfer of gene from white fly from plant to the white fly and allowing the white fly to survive on a different host or different plants.
So, while culturing Phyllanthus in our home lab, not only the sleep-wake cycle, but we also found another different things, that is insect-plant interactions, we can say. Um, So here, when in my home lab, I was culturing Phyllanthus to study its sleep-wake cycle, but I found a caterpillar feeding on my Phyllanthus plant. And it was very amusing to me as I didn't knew that Phyllanthus can be a host plant for caterpillars. So I tried to culture this caterpillar in a container, and but that time I did not have enough Phyllanthus leaves. So while discussing in chatShaala, I got to know that we can feed this caterpillar with pea plants or cabbage leaves. So I tried feeding that caterpillar with pea pods and cabbage leaves. So I tried to feed it with the pea pods and cabbage leaves, but it didn't survive in it. So that was a rare caterpillar, I thought. But again, in our college, there was some practical examination going on, and in that we have to identify Phyllanthus leaves. So that time I again got the same caterpillar in my Phyllanthus leaves. So that time I kept it in a container and brought it home. And that time I tried to culture that caterpillar with those Phyllanthus leaves only. I didn't give anything else to it. And this caterpillar turned into a beautiful moth, which is also known as Podoptera liturum.
So I have this Phyllanthus plant since 2020, but till that day, Sakshi observed I didn't. I don't want such things in my Phyllanthus plants.
Welcome to
First,
Cube
I
Home
want
Lab
to study
Chirikad, Kozhikode, Kerala. I too found such a caterpillar.
Okay, so this after Sakshi's observation, I also found there are caterpillars that we can also found in this Phyllanthus. So this is the one of the caterpillar that I got in Phyllanthus. If we look critically, how sweet or how cute they are.
We don't have to look critically, it is very visible.
So they are enjoying their food. And I collected that caterpillar and I put it in a pot. And I also didn't know that they are shedding their skin. So this is the shedding their skin. And also like silkworm. I also thought that is that a silkworm I got, but is that not a silkworm? I also observed that they are making a cage like a thing by using their skin. And also finally that caterpillar changed to a moth. And later also I observed there is another kind of caterpillar that is there in the Phyllanthus. Actually I got four. Out of four, two only emerges into moth. So here also we can see that caterpillar is so happy that is getting this Phyllanthus plant. He's also having some thorns, skin like things also there in there. So not only leaves, they are also eating the fruits and of these Phyllanthus. And finally that caterpillar changed a few pairs, then two more. So by collecting these caterpillar, I also mentioned I got four such a caterpillar, but out of four, two only emerges into moth. I didn't care properly, and also I forgot to cover once that pot. Then also I identified that caterpillar there.
This is the Phyllanthus plants that get Sushansa from Assam. So we can look the journey of Phyllanthus from Assam to Mumbai.
Go ahead.
There are more videos.
Move on.
I think this is a very interesting study, a very interesting model system. And through this model system, we can touch various fields of science, like we can see in the slide, such as seasonal studies, geography, taxonomy, experimental methodology, pharmacology, and Nobel Prizes are also given to study on studies on circadian rhythm, which QBs are trying to study on this particular plant. So I think we all can collaborate and study this interesting plant.
Thanks to all collaborators.
Thanks, it's a nice presentation and now, any question from the audience? Sir, please.
You had made a very bold statement that sunlight does not influence closing and opening of the leaves. So then what influences?
Light is not. Like we are trying to here observe the time, the sunrise time and the sunset time. We are is that time comes the leaf opening or leaf closing. That part we are trying to observe by those. Yes.
So
ma'am. Yes, sir.
sunlight time, sunrise and sunset timing, that only influences the sunlight.
But
So the time of sunrise will influence the amount of light which will be there.
Sir, the Phyllanthus plant leaf opening at a day and closing at night. So we thought that the sunrise, we have a hypothesis that the sunrise time comes later the leaf opening or the before the leaf opening. That is we want to find. So we observe that the sunrise time is 6:30. We can think of before 1 hour also the leaves starts opening. So our conclusion from this study is that the time, the sunset or sunrise time is not the factor. The time, if the time comes, the leaf opening and the closing, that is not the factor. Just before 1 hour, we are seeing that the leaf is trying to open its leaves. So
Because the sun is about to rise. So it has started opening because the sun is about to rise. Okay, so you need to revisit that and just take care.
Sir, there are some.
sir, and also that from that we want to conclude that not concluding, we want to say that the time, other than the time, there are other factors like the internal mechanism also there to control this circadian rhythm in plants.
There is one more question from Hina ma'am.
Yes, ma'am.
Very nice presentation, the entire team. But I have a question. How do you say your plant is sleeping? And you said that you're talking about the sunrise time and sunset time. What happens at sunrise and what happens at sunset? The light period only changes. So can you please explain?
Sorry ma'am, if we use the term sleep, so we are trying to analyze the circadian rhythm associated with their leaves. Whether their leaves is opening or closing. So ma'am's question is like, ma'am can you please repeat the time? Chandra sir, can you please give the mic again to ma'am?
How do you say the plant is sleeping? The leaves are closing, that means it's sleeping.
Okay sir, if we use the term sleep, if any one of us use the term sleep, sorry for that, we are just to convey the rhythm.
The entire label of your project is sleep-wake cycle. Okay, you're talking of the plant sleeping and waking. That means the entire time when the plant's leaves are closed, they are in the sleep mode. What do you mean by that?
Okay, so sorry for that if you use the term sleep. So while what we want to understand is that circadian rhythm. When we look at plant, we can see the leaf is opening and closing. So we came to know the concept of sleep and wake just before that discussion, that is the cursory were going on. So we can't say that is a sleep or wake. So we also thought that is the leaf is opening is a wake up and the leaf is closing is a sleep. But later we try to analyze the terminology. We can't use say sleep or wake. Instead of that, we came to know that the suitable term that is for circadian rhythm. The rhythm is associated with movement. Rhythm is associated with the leaf movement.
We have more questions online. It is from Vivas again. So, what is the importance of studying the circadian rhythm in phyllanthus? Why circadian rhythm is important in plants?
So why circadian rhythm is that we cannot see like when comes to other plants we don't able to see such kind of behavior like opening and closing like in a it is not visible to us. They may be having some other rhythms, but I don't know like we don't know that such kind of behavior we can see with our visible eyes is in case of this phyllanthus and in case of pea plants also they are showing and like we show it in mimosa, but in mimosa it's not the it's not that when we touch they close, but they are also showing this opening and closing behavior. So it is visible to such kind of plants. So that's why we choose this phyllanthus. So it is since it is easily visible to us, so we can study the behavior of plants, how they behave like that.
Yeah, one more question from Jay also.
See, I have that question because you are studying basically light, maybe if I use the term light effect on the plant opening and closing. So, what will happen if you artificially keep the phyllanthus like in a different light, like keep it in a dark for continuous dark period, continuous light period? What do you think will happen? Will the pattern change?
Yes,
Have you planned any experiment by controlling the light exposure to the plant?
Yes, sir. The cubists that done the experiment, like continuous light experiment, like Shraddha, Chitra ma'am, Sunita ma'am has keeping the plants in the continuous light. We have done it in our home lab. So by providing LED light, so in a room. So in that also we observed that even in continuous light they are opening and closing, but there is a delay in the there is a shift that suppose the plant is open at 6 a.m. and close its leaves at 6 p.m. But there was a delay of 1 hour or 2 hour in their opening and closing behavior, and they also noticed that after about 5 days or 4th day after they started seeing that the leaves are not completely closing. We don't know how it could be what changes that happens to them.
But like if you, for example, if you keep in continuous dark, have you seen in continuous dark? Because if the pattern changes, what is the outcome of it? Like what do you interpret? Or will the pattern remain same? Because if the it will help us to understand that whether the rhythm is internal to the plant, like it is genetic, or it can be changed by the environment.
So since the first 3 days of observations, they are behaving like opening and closing are happening. So even in presence without the presence of darkness. So so there is a internal mechanism will be regulating. There are some genes like in Drosophila also that found like that the period and timeless genes.
So will it be fixed? Because it will not change. Even if you change any light condition, it will be same. Pattern will be same. So that
Yes,
is totally genetic. It is like hardwired.
Yes.
And it is internal.
Yes.
And if it changes, so it means that it is based upon the external condition.
Yes, I think we need to more look that.
You can design some.
Lahari from Lahari wants to see the slide again, the Andhra Pradesh observation slide. Please, Kiran.
I think
Kiran, please show the Andhra Pradesh observation slide.
Lahari is one of the presenters of the Philanthus model
Yeah,
system. So when she was speaking, she was not audible. So at that time, she said.
It's okay. But I think this is the last question from Hina.
Lakshmi, you said that you only observe this kind of rhythmic movement in Palanthus and Mimosa. So when we talk of morning glory and certain flowers which open at certain time in the day and afternoon, this kind of rhythmic movement, this kind of movement has to do with light intensity in them too. And the pollinators visit them. Can you please throw light on this? Like you just said, I'm just asking that you said that you don't see any kind of movement. So the opening and closing of buds in certain flowers of morning glory, best example is that. So can you please explain your statement?
Yes, ma'am. It's not actually, the rhythm is not only in leaves, we can observe, like ma'am said that flowers, like 4 o'clock plant, and also their leaves are also like showing the behavior of opening and closing of the leaves. And there are other plants also, and also the opening of flowers is also as a guardian rhythm. That I
Okay, thank you. And I think it is the last question from Preeti. Yes.
So we observe that moths are choosing phyllanthus. So just how butterflies are choosing curry leaves, is phyllanthus being specifically chosen for moths for egg laying?
Yes, ma'am. So I do think like that. Because when we got this moth, we also search for is there any other host butterflies or moth is the host plant for this caterpillar is a host for this phyllanthus plant. So we found that there is a published article, I guess, that is sent by ma'am. I don't Hina or someone in the chatShaala discussion might be discussing about that. We got that there is one more moth is also a host plant for this phyllanthus. And they also published that paper also. So I think that there is much like butterflies. The female moth is also choosing some specific plant for laying its egg, and we can also test that by putting some other leaves whether they are able to eat or like they are surviving or not when we changing the plants that we are giving in the pot. In that way, we can also test whether the caterpillar is choosing for such plants.
Thank you all. It is a very good, very nice presentation. A big applause for them. Before finishing the session, please don't go. I am taking the opportunity. Because I will not have the opportunity next time after lunch. So as they show the train left here, and we want to continue the train left till 10 degree. 27 to 10 degree. So I'm taking the opportunity to hand over the two phyllanthus plants carried here from Assam 27 degree north and 95 degree east.
So that's a gift
So
for us.
I'm taking the chance to hand them over the phyllanthus plants. Kashyap, bring the plants. And I'd like to hand over this to Deeptha and Lakshmi so that they could continue the study. Yeah, you can't actually do that.
Out of the polythene.
Take a photo.
Someone should take a photo so that they don't photo award.
Why I took it as an opportunity? Because if I get them unofficially, they might have lost. Now they can't. They can't.
Now if they feel you can ask questions. Now if they lose, they kill the plant.
Yeah.
They're culprits. So the train journey would continue to Kerala.
Yes, very important. Thank you. Thank you. And I hope they will be live there. Yes, thank you. Now, we are going to end the session here. Please take it to the plants.
Lunch,
So today we are going
Yes.
shortage of time now. So we have four presentations till now. The first, we have art form presentation, then geographies, mosquito, and at last philanthropy in the first half of the day. Yeah. So people are hungry, they become hungry. So they're asking me to finish the session now. So we got a very wonderful sessions till now. We come to know how our firm has the same ganglion, same pattern of neurons like us. Then again, the regeneration study can help us to solve many problems. And in case of geographies also, you see the transparent eggs, transparent embryos there, very, very mosquitoes and still the process of malaria-like diseases. Yeah. We will collaborate that also. And at last, before coming to Cube and after joining Cube, we heard many times about finances from Professor Arunachal. Every time he said finances. That's why people all around the country in India, we are all doing finances started in rhythm study. So it's a very wonderful session today, and I'd like to invite you all to join the next session on the ground floor at room number 005. 001 to have the great session of lunch. Thank you. Thank you all.
Okay, please note that we just have half an hour because within half an hour we will please come back over here and we'll start with the next.
You're not coming back here, right? You're not coming back here.
So yeah, for the awards.
Are we coming back here?
Yeah, so we first, after lunch, we usually do a group photo. Yesterday's group photo. Yesterday's group photo shared before, but we usually have a group photo.
सबको अंदर पूछना
150, just remember the time, 150 years.